Hello, hello, welcome everyone to the second day of the ESCCA 2021 Virtual Congress. I hope you have had a nice day. Yesterday we did have it. It was very nice to start this Congress, and well, we really enjoyed having your companion yesterday in our room. Just let us go through what we saw yesterday. So we started the Congress by having a few presentations regarding Infinicyt, regarding Omnicyt, multidimensional analysis, the demonstration about Omnicyt, also some more detailed talks about prognosis of AML, analysis of monocyte subsets, also T-cell immunotherapy research, and then we had a closer session given by me regarding the EuroFlow panels. And well, today we have some of the presentations will be the same, and others will be quite new. To start with, I'll start just with a brief introduction to Cytognos for those of you who don't know about the company. But then we'll have quite a nice session regarding COVID-19 immune monitoring of COVID-19 patients using the primary Infinicyt Orientation Tube developed by EuroFlow within the immunology panels. Then we'll have again the multidimensional and spectral analysis using Infinicyt. Then we'll have the plenary session regarding multiple myeloma reporting by my colleague Maria Rodriguez. That session will be, I'll try to put it in this room, but it will be in the plenary room number 3. And well, we have it here. So it will be in the industrial panel presentation number 3, one of the plenary rooms, okay? And then we'll have the second online demonstration where 2 of my colleagues, Maria and Arantxa, will be taking care of the Omnicyt, and we'll be showing you for 1.5 hours the benefits of using Omnicyt, how it works, how you can actually acquire a few samples. They will be running a compensation. They will be acquiring an LST sample. This is the lymphoid screening tube. And well, of course, they will be glad to solve any question that you might have. Later, we'll have the session, your cheat sheet for smart clinical flow cytometry testing. And this is a session with a few tips and tricks regarding Infinicyt and regarding Omnicyt. Well, regarding flow cytometry in general, sorry, this is not focused on Omnicyt. But it covers also a little bit about EuroFlow. Then we'll have that session, about Omnicyt, regarding the full or the complete solution of using Omnicyt for high sensitivity studies in multiple myeloma. And then we'll have again that session focused on T-cell immunotherapy research, and the last session regarding the EuroFlow panel, Cytognos, whatever you feel, if you have any question or comment. Of course, I'm Jesus again, and my colleagues Maria, Karina, Arantxa are available in the chat here or in our virtual stand in case you have any comment or question about our products, about our solution, about Infinicyt, Omnicyt, whatever you feel like talking about. Just let us know, and we'll be glad to do it with you. Another thing is that all these sessions are recorded. The session yesterday was recorded for like the 7 hours from 10 to 5 was recorded in a full session, a complete session, and that will be available hopefully here in the ESCCA material. So that will be available later, and you can look for any of the sessions that we do during these days. And maybe we'll also be able to upload that in our website. in separate content, or we'll probably let you know about it either by email, or we'll publish that on our social media and LinkedIn. So yeah, again, if you have any comment, question, don't hesitate to let us know. And now I would like to start with this session regarding Omnicyt, or regarding, sorry, Cytognos about us. So let me close this, and I will start with this. So Cytognos, who are we? Well, Cytognos is a biotechnology company, and we are specialized in clinical flow cytometry. We offer complete solutions for three main purposes. These are oncohematology, that's the first one. The second one is clinical immunology, and then we have clinical trials. Our goal actually is to help, as stated there, the flow cytometry labs to provide better patient care and more efficiency workflow and a more efficiency performance. And we do that through standardization, using efficient tools, using reproducible tools, and also being highly sensitive. So with these four elements, we are able either to provide better patient care or to improve the laboratory efficiency of your lab. Of course, we are mainly focused on the clinical flow cytometry lab, but well, as you know, we are indeed focused on very innovative solutions for all these three pillars: oncohematology, clinical immunology, and clinical trials. We are not focused on, let's say, basic or classical approaches to flow cytometry analysis. So yeah, at the end, both for clinical labs and for research labs, some of our tools will be very useful. We know that, and it has been proven by thousands of clients that use our product, our reagents, software, and medical devices all around the world. And well, of course, this is not just because of us. We are not the only owners of all these, let's say, gains that we have achieved throughout the years. This also obtained thanks to the collaboration that we have with international universities, with research centers, and many other companies that help us, and we collaborate in order to obtain the best tools and to provide the best tools for our customers. Okay. So I would like to give you a few insights about our history. Cytognos was founded in 1996 as a spin-off from the University of Salamanca. It was founded within the hematology department, and of course that's basically due to our initial focus on hematology. This was when we started introducing some of our reagents, single antibodies, combination of antibodies, and it was a few years later, in 2006, that we started our collaboration with the EuroFlow consortium, and this was indeed one of the key milestones in our history. EuroFlow was founded as a scientific consortium this year in 2006, and we were one of the initial companies that joined the consortium together with other laboratories, research laboratories from different places, from different points in Europe. At that time, it was around 10 to 12 labs, and the aim of EuroFlow was basically to enlarge or to develop a more profound knowledge and also different approaches and methodologies for the analysis of hematological malignancies through flow cytometry. So again, some of the pillars were the standardized approach, to analyze statistically and through let's say real research not personal experience, not opinion, but thanks to data, to develop these validated panels, and also not only to develop them, but also, as I said, just to validate them in order to ensure that the performance and their usefulness was convenient for the clinical perspective. And then at that point, we were also in charge of the development of Infinicyt. So Infinicyt is our proprietary software that we develop as a company. And well, as you know, this was a huge development and a huge theme for Cytognos. That's why between 30 to 40% of our employees are software engineers who are developing Infinicyt, and that's why we have sold more than 1,000 licenses over the years, and our software is being used all around the world in clinical flow cytometry. A few years later, so EuroFlow started the research around 2006. They developed their methodology, the standardized methodology, the EuroFlow panels, including the best markers and fluorochromes and the best combination of antibodies for some of these, for some diseases. And then in 2012, we started the commercialization of some of our lyophilized kits. So basically, we had been providing already some other kits and combination of antibodies for other purposes, for other diseases, different combination, of course. But it was at this moment that we started providing lyophilized kits, both based on our own research and the developments of EuroFlow. So that's when we introduced the LST kit, the lymphoid screening tube, the ALO tube, the SST, the PCST, and so on, right? And well, the standardization of the reagents brought to another step in the standardization that happened in 2017. And that's when we introduced in Infinicyt, 10 years later, after we started developing the software, the automatic gating and identification tool. So this is the software feature that allows the user, the flow cytometrist, to automatically analyze a clinical sample based on the comparison of the sample with the reference databases built by EuroFlow. So basically, with Infinicyt now, you can take your FCS file coming from a patient with a suspicion of acute leukemia or a multiple myeloma. You can put it there in Infinicyt, load it, the database will analyze it. You will just review a few things, and you will obtain a report that will be helpful for you. Like, it will be providing you useful information for your analysis and also for the clinician, because it will include some clinical insights regarding the frequency of abnormal and normal populations, the hemodilution of the sample, for example, in the case of multiple myeloma, the abnormal markers of an abnormal population if present, and so on. So there are a few elements which are very important here. One is the standardization of the process. So now you can just take a sample that you took or acquired from the cytometer, no matter whether you are an expert or you are not so expert, whether you are an experienced person with this type of analysis, let's say multiple myeloma MRD analysis, or whether you don't know much about this analysis yet, you can load the sample. You will do exactly the same process, both person, and you will be able to review the sample with the help of the software. You will check these populations, so at the end you come to the same result and basically the same conclusion. So that's one thing, the standardization of the analysis. The second thing is actually that we also standardize the reporting. So we built this report not just based on our preferences or the preferences of some of our colleagues, but indeed the multiple myeloma MRD report, as in the same manner that other reports in the automatic gating, are built taking into account the consensus, the scientific consensus, and the clinical requirements, so that actually all this communication between flow cytometrist and clinician is standardized, and actually we provide everywhere the same kind of information. So that will be good for the sake of improvement and standardization all around the world. 1 year later, we also launched our first medical device. This is Omnicyt, our CE-IVD flow cytometer. This is our flow cytometer with 3 lasers, up to 4 if you upgrade the instrument. In the basic configuration, it has 14 parameters. It has acoustic focusing that provides, and also volumetric acquisition that provides more reproducible and more consistent measurement of your cells. No matter whether you are acquiring at low speed or high speed, you will obtain a very homogeneous signal, and that's also due to the flat top lasers that avoid misalignment of the lasers. And well, it can acquire a very high speed because of the volumetric and the electronic system, up to 35,000 events per second, which is quite high compared to other instruments. And well, with Omnicyt, the important thing there is that it's a CE-IVD instrument, fully validated and certified for the 3 lasers, and indeed it was validated by acquiring samples from the LST, the lymphoid screening tube, and the multiple myeloma tube, so the multiple myeloma panel. So yeah, Omnicyt was validated in the real clinical and hematological practice that we are focusing on. And well, it's been quite a good 3 years for the moment. More and more people is using Omnicyt across Europe now, and we really hope that you can take a look at it later in the demo session. I remind you that this session will be taking place at 12:30 approximately, and from 12:30 to 2. So it will be 1 hour and a half where my colleagues Maria and Arantxa will be explaining you all about it. If you are not able to attend this session, just remember that you can see the recording later in the ESCCA material, hopefully, or in our website. And you can also request a private session for an Omnicyt demo, online demo with some of our specialists in our website. Okay. 2 years ago, we opened our new offices in the USA and UK. These are 2 of the countries where we are serving our products directly. So my colleagues Irene, Dan, and Chris in the USA and UK and Ireland respectively, they will be glad to hear your comments, to receive your questions or your support, and we'll be glad to support you in order to provide you with our solutions. By being there and present in your region, we hopefully will provide a much better customer support and also a commercial experience for you. So don't hesitate to contact us, and we'll be glad to help you. And then just in the last few months and years, of course, the last few months have been quite a different experience for all of us due to the pandemic. And well, one of the key things that we have been doing the last few months is to develop or to further increase our offer to address the COVID-19 pandemic. We developed a few solutions that are able to help you in the analysis of COVID-19 patients and other elements. There are mainly 2 pillars here. One pillar is the immune monitoring of COVID-19 patients, so you can actually see what's the immune status of the patient right after COVID-19 infection, then for example 2 weeks later and a month later. And what we found is that you can clearly see a difference between the presence of different populations, depending on the timing of this measurement. and that actually can provide insightful information on the progression of the disease. And then the other element that you can analyze using a different type of study is this study, the Oxford study, and with that you can actually measure the response to vaccination. You know, vaccines are quite a topic these days. With this study, Oxford, you can measure the response of our T cells to vaccination, and well, yeah, you can measure how right after vaccination there are some level of cells, and then that changes throughout the weeks and months, and they are still, of course, these studies are still in progress to see whether there is a real immunization of the system or not, or whether some people will respond differently to the disease. And then also just a few months ago, we released the FDA 21 CFR Part 11 Infinicyt software tools. So this means basically that we included some user access control features that allow you to have different users for Infinicyt with different passwords and different write levels, so that some users can actually analyze the samples, others can only review them, and they cannot modify the data to each other. And it's very important for regulatory purposes, not only in the US, but also in Europe. So check it out there, and don't hesitate to let us know if you have any comment or question about that. We are in Salamanca, so Cytognos was founded in Salamanca, and that's where our main headquarters and offices are. Most of us are working from Salamanca these days. Some of us are working from home, and also our commercial team in Madrid and Portugal is they're working from Madrid sometimes. But yeah, Spain is where our origin is located. And as I already told you, we have another 2 offices in the UK and USA in order to serve our clients in the USA and UK and Ireland. Then we have another branch office in the Netherlands, where my colleague Maria is assisting customers from the Central European region and North Europe. And of course, we also want to thank all the distributors and our network of commercial partners who are serving our clients all around the world. We are present in more than 50 countries, and well, they are of course the core or one of the pillars of our business that ensures that we can serve as many clients as we do at the moment. We renovated recently the facilities in Salamanca. I just brought this picture just to show you how nice they are. We really love them. The new conference rooms, the new reception, everything is quite new. This is not only that we renovated the offices, but also that we built new facilities, because of course the team is growing significantly. And well, this also includes both a new lab where the antibodies are either manufactured and the quality control is done, and so on, and also the inventory management. And then also we have a new kitchen, which is very nice, but of course we cannot use it that much these days due to the pandemic. Our team is composed by more of 75 employees, some of them or several of them including PhDs. And well, there are of course different departments, the product specialists like me, who are trained in clinical flow cytometry, quite specialized with deep technical knowledge, not only from the technical perspective of flow cytometry, but also from the clinical perspective. And we are trained significantly at the University of Salamanca, one of the main research and academic institutions in Spain and Europe, one of the oldest universities in Europe, and also with a very important core for flow cytometry, as you probably know. Our sales network it's present both at national and international level, of course, and then around 3 around 30 to 40% of the team, as I already said, is probably composed of software engineers who are developing Infinicyt and make sure that the software is updated every day. It's continuously improving, and we keep adding and adding more features in the future to Infinicyt. Our lab technicians are in charge of the research and development, our innovation, manufacturing, and quality control. And of course, that's very important too, because our portfolio has been growing throughout the last years, both from the basic perspective of single antibodies and other reagents, and also other innovative solutions such as lyophilized kits, combination of antibodies, new panels, and so on. And of course, our wonderful administration staff who is doing accounting, regulatory affairs, shipments, administration tasks that really help us, and allow us to really work as a team. So, well, with that, I would like to finish this introduction to Cytognos. And just for you, I would like to show you also, just a second, I would like to give you a little tour through our website. Let me just a second here. So here, if you open cytognos.com, in events, upcoming congresses and meetings, you have here the ESCCA virtual conference. It works too if you just write cytognos.com ESCCA 2021. That works too. And then you have here all the information related to the ESCCA meeting that is taking place. So here you can see the schedule of our presentation that will be happening in just 2 hours, 1 hour and a half by Maria. You can also download some of our brochures, some white papers, and also the new EuroFlow posters, which are very nice. You have also access to our virtual sponsor room, the schedule, and you can see here the schedule that we are and we'll have today. And as I said before, you can request your own Omnicyt online demonstration if you want. So this is a private demonstration that you would have with some of our colleagues, 1 hour. Basically, of course, we are flexible there, and it will be covering the basics of Omnicyt and a practical demonstration, including both compensation and acquisition of the LST. You can request your Omnicyt demo here just by filling in some data. And finally, just remember that we are participating too in the ESCCA raffle, and we are giving away a full year Infinicyt advanced license, including 200 EuroFlow database accesses. So if you have any comment about the test and the questions that we include in order to participate in the raffle, just let us know. Well, we tried not to put them very hard, but yeah, I hope that you can participate in the ESCCA raffle and you can win the license. I think that it will be wonderful for you. So now we'll just wait a minute and we'll start with this session, immune monitoring of COVID-19 patients using the PID Orientation Tube. Just as a reminder, all the sessions will be available later on demand. So this session is being recorded at the moment. And some of the sessions that I will be watching today are happening live, others are recorded, okay? So if you have any comment or question, just write down in the chat. We'll be glad to help you. If you are watching this later, recorded, just don't hesitate to write to us, and you can do that directly here in contact. So yeah, here you can contact us directly. So if you have any question, just write there, and we'll be glad to help. So let me just now stop sharing my screen, and I will put the video just now, going to the second session. Okay. So we said that the first session will be this talk by my colleague.
My name is Javier.
Javier. and he will be speaking about the immune monitoring of patients with COVID-19 and how our primary efficiency orientation tube is useful for that application. I will just share the talk now, and this will be around, I think, for around 40 minutes. And then if you have any comment, if you have any question, just don't hesitate to write in the chat or to write to us, and we'll be glad to help. Okay, let me know if you cannot hear the talk, and everything should be fine.
My name is Javier, and I will be presenting today's webinar about Cytognos next generation flow solution for immune monitoring COVID-19 and other pathologies using the PID and the immunoglobulin subclasses kit. First, let's talk about the value of the clinical lab. The clinical lab plays an essential role in the healthcare system, as it provides the information that enables doctors to make the right diagnostic and therapeutic decisions for their patients. So, as clinical laboratory professionals, we have a direct impact on many aspects of the patient care, including making a direct improvement in the life of the patient. And of course, one of the fields where clinical labs have a key role is in the monitoring of patients. In order to provide this high-quality information, we need to address several aspects that have to do with mainly two things. In our labs, we always look to improve our laboratory efficiency, and for this, we will try to optimize resources, ensure the reproducibility of our instrumentation, minimize the subjectivity of data analysis and reporting, and having a good training program in place for our lab staff. And also, we always have in mind that our work will provide better patient care, as by providing a timely and accurately laboratory testing, we get a quicker diagnosis that leads to faster and better treatment, which eventually translates into shorter hospital stays for our patients. With this in mind, from Cytognos, we work to help clinical flow labs in achieving these aspects, both providing a better patient care and being more efficient in their workflow. And that has been our goal while developing flow cytometry solutions. After this brief presentation, in today's webinar, we will review the role of immune monitoring in pathologies such as cancer, autoimmune diseases, and infectious diseases. And here, we will focus on COVID-19 and will analyze how flow cytometry can help us to better understand this pathology. We will move on to our next generation flow solutions for these applications, where we will present you the PID and the immunoglobulin subclasses kit, oriented as part of this complete solution. And I will show you a few files, including a patient file, as an example. We might also have some minutes at the end for a couple of questions. Okay, now let's move to immune monitoring patients. We know that one of the traditional divisions of the immune system could be as lymphocytes, where we can find B, T, and NK cells, and myeloid cells, where granulocytes, monocytes, and dendritic cells can be included. Within these groups, we could identify more functional groups and have a deeper knowledge of the distribution of these subpopulations. But we also know that these are not closed groups. All the different immune cells interact with each other, exchanging information about the environment and regulating the function of the rest of the cells. In this sense, if any of these cellular compartments is affected, for example, in the case of an infection, the other cells in the immune system may be affected as well. And that's going to change the homeostasis of the immune system. That's why it's important not only focus on the target population of the disease that we are studying, but to analyze the environment so we can know the status of the immune system and obtain information from it. And in this sense, immune monitoring our patients before, during, and after therapy will provide us with a general view of the immune system and will give us details on how the different populations can be affected. And this can be especially relevant in cases of immunotherapies, as most of them affect directly to the immune system. It also allows us to track the recovery of the immune system over time. And this could be very important in some diseases, as there are studies correlating a normal recovery of the immune system with a better prognosis of the patient. For example, after autologous bone marrow transplantation. We can also follow the pathological population after the treatment has finished and compare it to pre-treatment levels. So, monitoring our patients will bring us several benefits. It will improve our knowledge about the state of the patient. It can bring prognosis value in the dynamics of recovery, and will allow us to make clinical and therapeutic decisions according to this knowledge. All of this in the end resulting in better and more personalized care for our patients. And as most of us here are cytometrists, flow cytometry is a very good technique for this purpose, making the monitorization very specific and reliable. There are more and more studies highlighting the importance of characterizing not only the target population, but also the homeostasis of the immune system. Here are just a few samples related to the fields of hematology and autoimmunity, as monitoring the recovery of the patient after treatment and knowing the tumor microenvironment can provide us of vital information about the response and survival of our patients. And from this point, we are going to focus on the immune monitorization of COVID-19 patients. We're going to make a review of some of the works that have been published that highlight the benefits that bring the monitorization by flow cytometry of COVID-19 patients. All the information that we can obtain by this technique, and also how can patients benefit from it. So here we want to refer to the review that was recently published by the group of Andrea Kosaritza, where you can find most of the articles that we are using in this webinar. First studies focus on the circulating immune system by flow cytometry in patients with COVID-19 were carried out in China, and they showed a significant difference in cell distribution in patients compared to normal distribution ranges. As you can see, for example, here with the total T lymphocyte count that is reduced in comparison with the normal range. On top of that, these changes were especially strong in severe cases of COVID-19, and the reduction of normal counts could distinguish patients with a severe form of the disease from more moderated cases. So, the first conclusion we can get is that the immune monitorization of COVID-19 patients by flow cytometry could help us distinguish between moderate and severe cases of the disease. These findings were reproduced by other groups, and in this work they correlated these findings with clinical parameters of inflammation. We can see how levels of inflammatory markers like the erythrocyte sedimentation rate, the C-reactive protein, and the interleukin-6 were higher in patients with low counts of T cells, and this correlation is stronger when referred to CD8 T cells. So as you can see, changes observed in the distribution in patients can be directly correlated with other clinical parameters. As we said before, monitoring over time can also bring us very relevant information about the progression of the patient. In this article, we can see that by tracking the immune system of COVID patients, we can observe that patients who recovered normal levels of circulating leukocytes were those who were able to completely eliminate the viral load, while the patients that after 10 days still presented reduced counts could not eliminate the viral load. This brings up the prognosis value of making immune monitoring by flow cytometry. In this research work, it was found that in lung aspirates of COVID-19 patients, the distribution of the cells were severely altered, and way higher percentage of myeloid cells were present in patients compared to healthy control. As in the previous works, this could also differentiate between severe and more mild cases of the disease. So we have to take myeloid cells into account to get deeper and better information about COVID-19. This work connects with the idea of the previous one, as here the group categorized patients using two parameters, the levels of circulating immunoglobulins and a neutrophil leukocyte ratio. Interestingly, they found that patients with a more severe form of the disease concentrated in the group that presented both high number of neutrophils and also high titers of immunoglobulin G, showing us again the importance of studying not only the lymphocytic compartment. Also in this same work, they also found that the dynamics of serum immunoglobulins were different between patients. As you can see here, in the more severe cases of the disease, they found that it was produced a peak in the concentration of the soluble immunoglobulin M. Now it's important to notice that not that one of the treatments proposed and that is already being applied in some countries is the infusion of hyperimmune serum of people that have gone through COVID-19. This infusion will make us unable to monitor our patient at serum level. However, by flow cytometry, we can monitor at cellular level, so even when we are altering the serum immunoglobulins, we can still check at the cells that produce these molecules in the patient. And finally, we'll come back to where we started, the review of Andrea Kosaritsa. After reviewing these different studies, the group pointed how we are still missing a lot of information about the disease, and we wanted to comment some of the conclusions they reached by the end. Like the role of the innate immune cells. As we have seen, very little is known of their distribution and how this knowledge could help us get ahead of the virus. Also, we need a better and deeper understanding of the role of the different subpopulations of CD4 and CD8 T cells. If the changes that we see in the patients are produced in specific compartments of the T cells. And also the kinetics of the humoral response, along with the importance of the memory B cells and plasma cells in the development and the control of this disease, as we also need to understand what happens not only during the infection, but also after it with the antibodies and the memory cells to prevent reinfections. With all this data in our minds, we can now discuss how our technique should be in order to successfully monitor COVID-19 patients by flow cytometry. As we have seen, we need sensitivity in order to be able to compare the data of our patients with a robust database of normal samples with normal reference ranges. It is very important as well that our technique is standardized and by so reproducible, as we intend to follow one patient over time, and we want our data to be comparable from one day or week to another. Ideally, we will need to identify as many populations of lymphoid and myeloid cells in an efficient way, without the need of a huge screening panel full of different tubes, so we can easily introduce this application into our lab routine. And as we have just commented, we should be able to detect expression or distribution changes at cellular level, so we don't have to worry about different treatments affecting our monitoring. And to cover all these aspects, I'm going now to introduce Cytognos next generation flow solution for immune monitoring. This solution will cover all the points we have just seen in order to facilitate the implementation of this new application in your lab in a way that you gain efficiency and you also improve the care of your patients. This solution includes antibody reagents, which in this case are going to be the PID and the immunoglobulin subclasses kit, standardized operating procedures developed and validated by clinical flow cytometry experts, the flow analysis software Infinicyt, and Omnicyt, or CE-IVD instrument. All of these provided with the training and support you need to get your assays up and running. One of the two kits that conforms this solution is the pilot kit. With this 8-color panel containing up to 12 markers, we will be able to get a complete identification and enumeration of the circulating lymphocyte and myeloid populations. We will see it in more detail in the demo, but with this panel, we will be able to identify most of the populations of circulating leukocytes. In a first step, we can identify B, T, NK cells, and also myeloid populations. And in a second step, we can classify the two main T cell subsets, such as cytotoxic cells using CD8 and helper T cells using CD4. And we will analyze the distribution of naive, memory, and effector cells in these two populations. We can also identify different B cell subsets, since this panel includes the marker to study pre- and post-germinal center B cells. And here, in the post-germinal center B cells, both unswitch and switch cells. And what is also very relevant, within the myeloid compartment, we can detect the different types of granulocytes as neutrophils, eosinophils, and basophils, as well as monocytes, both classical and CD16 positive. And finally, we can identify the plasmacytoid dendritic cells, having a very detailed landscape of our myeloid cells. With this panel, we are identifying over 20 different immune populations in a very efficient monotube panel. And also, with all the markers present in the tube, we will be able to detect any change that is produced in the patient at cellular level, which will give us the information we need to make better clinical decisions. We also talked about the importance of having a robust comparison to detect changes in our patients in a sensitive way. And to address that, as we previously remarked, our solution is based in the work of the EuroFlow group. With our solution, using the PID panel, we can compare the data obtained from our patients with the published normal reference ranges that were developed by EuroFlow. And these ranges are presented specifically segmented by age. This can be very important, as we all here know the distribution of the different populations of the immune system change throughout our life. And we can have different number of lymphocytes and myeloid cells compared with a child or with an older person. And so we are going to compare every and each population that we identify with our panel with their normal reference range. And for some population, as the plasmacytoid dendritic cells, these ranges could be very difficult to find. All this will avoid the need of validating and developing our own control population, making it a very reliable and fast to implement technique. Also, you have all this information published with open access if you want to check it or go deeper into these papers. Another advantage of our solution is that working with the Infinicyt analysis software, the access to this information is very fast and intuitive, as using the automatic gating and identification tool of Infinicyt, we have access to the automatic report. This report will present us the relative distribution and the absolute count for all our different populations, and we can compare it with the normal ranges of their specific age. There are 14 age ranges to cover from childs to adults, and this will allow us to make our comparison in a very precise way. This report also highlights the relevant information found in our patients, making the transmission of the data very efficient in our lab. As you can see, for example, here with this post-germinal center population, it is highlighted because the absolute count of these cells is reduced compared to their normal ranges. Now, to see all this in action, Let's jump to Infinicyt. We are going to start with a normal sample. So I can show you all the population that we can identify with the PID panel. This sample, as you can see, because all the cells are already colored, is already analyzed using the automatic gating and identification tool included in the Infinicyt software. This tool compares our sample with the EuroFlow databases and automatically assigns all the events that matches with normal populations present in the database, making our analysis faster as we just have to focus on analyzing the events that are different from normal cells. You can also see that the software presents us some multiparametric diagrams, and these diagrams will allow us to check in a simple glance if we lack any of the populations in our sample. For example, here it's presenting the different lymphocyte subset that we can find in the sample. Here will be the B cells, NK cells, gamma delta negative T cells, and gamma delta positive T cells. So we can easily see that they are all present. With the automatic gating and identification tool, our objective is to make the analysis more agile and bring the important information to the front. Now, we're going to go through all the populations present in the sample, and I'm going to start analyzing the lymphocytes. For this, we can start using this plot of CD3 and CD19. And as you can see here, we can identify our T cells and our B cells, as well as inside the T cells using the TCR gamma delta marker, we can identify this subpopulation of T cells. And this group of cells that are negative both for CD3 and CD19, and that are positive for CD16 and CD56, are going to be our NK cells. Going deeper into these populations, and starting, for example, by the B cells, using only these two diagrams, we will be able to identify all the different populations that we can identify with this panel. Using the marker CD27, we can distinguish between the present pregerminal and postgerminal center B cells, as pregerminal center here in pink does not express the CD27, while the postgerminal center B cells express this marker. And here, in this diagram, that we have immunoglobulin D and immunoglobulin M, we can study within the postgerminal center populations which one have gone through the class switch recombination. In this case will be these orange cells, as they have lost expression both of IgD and IgM, and the unswitched population that still maintains the expression of IgD and IgM. We can also detect a small population of B cells that are CD27 positive, but that only express and very highly IgD. Moving into the T cells, with this plot here, we're going to identify the two major populations of T cells. As we can see here, we're going to have our CD8 cytotoxic cells and the CD4 T helper cells. But we're not going to stay there. As here, using CD27 and CD45 array, we're going to be able to identify the different maturation populations of T cells, both for CD4 and CD8. For example, here with our CD4 T cells, you can see that we have our naive T cells that are CD27 and CD45 array positive, our central memory or transitional memory CD4 T cells that maintain the expression of CD27 but lose the expression of CD45 array, and our two effector populations of CD4, the effector memory, and the effector CD4 T cells that are differentiated by the expression of this marker. We can check that in the CD8 T cells, the pattern of maturation is similar, but we can find some different populations here. Same as with the CD4, or naive CD8 T cells are going to be positive and bright for CD27 and CD45 array, and our central transitional memory CD8 cells are negative for CD45 array, but still maintain the expression of CD27. And here, in our effector populations, we're going to have the effector memory CD8 T cells and two different populations of effector cells that are differentiated by the expression of CD27. If you remember, one of the points that we found important in the review of the article was that we needed a better understanding of the subpopulation of T cells. Sorry. And with these markers, we can check if there are changes in the distribution of the memory and effector T cells. So now let's move to our myeloid populations. And we can start by analyzing the different subtypes of granulocytes. As with this panel, we can identify eosinophils, neutrophils, and basophils. And you can see them here by this CD45 against the site scatter, where we can clearly differentiate them, as well as using the CD16 marker that clearly identifies and separates our neutrophils from the rest of the granulocytes. In addition to granulocytes, we also commented that we can check the different populations of monocytes, the classical monocytes or the conventional monocytes, and the CD16 positive monocytes. And they can be identified here very, very clearly by using this plot of CD45 and CD16 or CD56. You can also check here that this population of CD16 positive monocytes are a bit brighter in the CD45 mark with the CD45 marker. And finally, we can also check for the plasmacytoid dendritic cells that are going to be in the area of the transition between lymphocytes and monocytes. They are going to be both negative for CD3 and CD19, but with a low expression of CD4, and they are also very bright. They express very brightly CD45RA. As you can see, with this panel, we are not only studying in detail the lymphocyte, but we also obtain very valuable information from all these myeloid cells, getting the knowledge that we need to better follow these populations in our patients. Now that we have finished the analysis, we can move to the automatic report that Infinicyt generates based on the results of our analysis. Here, as you can see, the report is presented to us in a very intuitive way where we can check the absolute count and the relative frequency of our sample compared to the reference ranges of their specific age range. And for this case, we can see that it is applying the range of 30 to 59 years. We can now use these normal ranges to make sure if our patient presents any alteration in the distribution of these immune populations. This is a normal case and all seems to be fine. As you can see, for example, if we take the B cells, the absolute count and the relative frequency in this sample are both inside the normal ranges for the specific age segment. But now let's move to a COVID-19 patient. I'm going to close this and open the COVID-19 sample. Okay, sorry. This is a sample from a COVID patient that was taken the day of the hospitalization. So we will call the day 0 sample. Please take into account that this is a single file from a specific patient, and that the differences that we find in this sample might not be present in others. Our goal here is to be able to find the differences in our patients. And to not make this very long, I'm going to focus in showing you some of the changes in the expression of a couple of populations. But you can see here in our column of alerts that the software is already noticing that we have differences in this sample compared to the normal database. But as I said, let's bring the B cells and analyze them. Here, in the plot of CD27, we can observe that both post-germinal center populations, the unswitched, and the B cells that have gone through immunoglobulin class switching are expressing very brightly this CD27 marker. This could mean that the B cells from this patient are taking a more producing-like phenotype and are converting into plasmablasts to start solving the infection. Moreover, the unswitched B cells seem to be focused on the expression of IgM, as we can see in the plot of IgD versus IgM. We're being able to monitor at cellular level the changes that we saw that are produced in this patient, and understand how are the serum levels changing from this point of view. Another change that we can observe is the expression of CD16 in neutrophils. As you can see here in this plot, CD16 seems to be reduced in a population of neutrophils in this patient. CD16 negative or low neutrophils are more immature cells that are being released from the bone marrow. It seems like the immune system is fighting against the virus and is bringing cells at a very fast pace. So now, let's move to the report to check if there is any alteration of the normal distribution of our population. Let's wait in, okay. Now, here we can see that some of the immune populations are highlighted, and this means that they are out of range, either in the absolute count or the relative frequency compared to the normal database. We can see, for example, that our pre-germinal center B cells, you can see here, are reduced, and that CD27 negative effector T cells in the CDA are augmented. You can see it here, they are augmented in comparison to the normal range. In addition to this, we can observe that the myeloid compartment is severely affected, as all the granulocytes, monocytes, and dendritic cells are reduced in this patient. You can see it here, for example, that they are totally out of the range and they are very reduced. So, as we saw with the review of the articles, checking these populations might indeed be very important. So let's move back to the presentation. And as we have seen with Cytognos solution, we can monitor very precisely the changes that occur in a patient. And these changes can be in the expression of the markers present in the panel, as we have seen in the patient sample, where the expression of CD27 is higher than in normal samples of the B cells population, and the reduced expression of CD16 neutrophils. But not only that, we can also check any changes that are produced in the distribution. Here we can see the report of the patient that we have just analyzed at day 0, the day of hospitalization, as we said. And we analyzed the same patient after 7 days in the hospital, when the patient was almost totally recovered. And as you can see here, with Cytognos solution, it's very easy to follow any change in the distribution of all the populations that we study with the PID panel. You can check how after 7 days, the number of CD8 T cells increased, and this was even more evident in the memory and effector populations. We also see that neutrophils were deeply reduced, and this maintains over time. However, monocytes have recovered their normal levels. And interestingly, in this patient, we can observe an important increase in the plasmacytoid dendritic cells and also the basophils. As we are now able to detect these changes, we can follow our patient in a very efficient and sensitive way, and that gives us the chance to improve how we manage COVID patients. At this point, we have gone through how the PID reagent is going to help us monitoring the patients. And now we are going to check the other kit that Cytognos offers for a deeper monitoring of the B cell compartment, the immunoglobulin subclasses kit. With this reagent, we are going to focus on the B cells, and we are going to characterize the different populations of immunoglobulin G and immunoglobulin A. Using this panel, we will be able to identify both B and plasma cells producing IgG1, IgG2, 3, and 4, and also IgA1 and 2. In addition to this, we also study the expression of IgM and IgD. And due to having these markers, as we have discussed previously, the identification of the cells producing antibodies at cellular level will complement the measurement of soluble immunoglobulins at serum level. And this will allow us to detect changes that are produced in these cells even when the treatment alters the circulating immunoglobulins, due, for example, to hyperimmune seroinfusion. To detect changes in the patients in a sensitive way, we also have available in open access publications the age-segmented reference ranges for both B and plasma cells that we can use to detect those alterations in the distribution that are produced in our patients and compare them with a control group of their specific age range. And now that we are approaching to a vaccine, the monitorization of the specific immunoglobulin subpopulations might give us key information in the way of making a viable vaccine with a strong and durable response. And also, as we saw with the PID, having these reference ranges available with specific age segments, it's very important in the case of monitoring the immune system, due to the changes of the distribution that we present throughout our lives. And so we will have a specific way to know how are our patients evolving. And just to sum up, what today we wanted to show you is how we can address all these key points using the next generation flow solution proposed by Cytognos of monitoring not only COVID-19 patients, but also from other pathologies. We have seen that with our solution, we can compare our patients with an age-segmented database where we have normal reference ranges that will make our detection of changes in the distribution very sensitive. We also discussed how, based on the EuroFlow methodology, with their SOPs as one of the central points of our solution, we get the standardization and validation needed to make our monitoring reproducible. And this will allow us to compare samples with other centers and follow our patients over time. And finally, we also checked how we can identify a large number of lymphoid and myeloid cells and detect changes in their distribution or in the expression of their normal parameters at cellular level in a very efficient way. This will optimize our resources in the lab and will allow us to provide the clinicals with valuable information to help them in their decision-making with the patients. My name is Javier, and I will be presenting today's webinar about Cytognos.
Okay, so I hope that you liked this presentation, very detailed presentation about our solutions for immune monitoring of COVID-19 patients using the PID tube. If you have, I have seen that there have been quite a number of people joining the room throughout the session. So I'm sure that there might be some questions or comments about it. If you are interested in getting more information about this solution, our primary efficiency solution, and also about the immunoglobulin panels or the EuroFlow immunology panels in general, please let us know, and we'll be glad to give you more information about it. Also, I would like to remind you just something. We'll be starting now the next session, which will be multidimensional spectral analysis with Infinicyt. I know that will start in half an hour, so around 20 minutes. So yeah, we have 20 minutes now in case you have any comment or question that you want to ask. But I want to show you something. Just give me a second. Because actually, I already explained this, that we have created a new poster, well, 2 new posters, including the EuroFlow panels. And here, these are the posters that include the primary efficiency orientation tube, the immunoglobulin panel that Javi was speaking about, and you see how in this poster, well, it gives you the rationale behind it and what's the methodology that you would use in order to diagnose the different primary immunodeficiencies. Of course, in the case of COVID-19, this is a different approach, a different methodology used, but well, it will be interesting for you. And the thing is that you can download this poster from our website. You can either go to our, let me check a second. You can either go to our stand and download the brochures here. If you just click here in the brochure, you can directly access the download brochure system. So here, this is the poster that I'm talking about. You can also have the EuroFlow workflow poster, a response to COVID-19, and well, it might be interesting to open that one just for you to see it. Okay, so well, as you can see here, this is the primary efficiency. This is the one that I was just showing. So if you want to download it, it's here, free, available. Then we have the EuroFlow panels. So this is the same poster, but for the hematology panels, including the screening tubes by EuroFlow, the characterization, and also monitoring of multiple myeloma and BCP-ALL. Sorry, I think I might have a question in the chat. Not for the moment. And yeah, so these are the new posters. These two we just created a few weeks ago. And then we also have this, let's say, white paper regarding our solutions for COVID-19. So here you will find interesting information, a very brief brochure about our approach for immune monitoring using the primary efficiency orientation tube, and also the immunoglobulins. And then the other pillar, I was talking about that before already, regarding analysis of the immune response to COVID-19, well, to the SARS-CoV-2 infection. And this is also related to the vaccination, right? So this can be used for different purposes it can be used to assess the prevalence of the disease, either, as I said, infection or vaccination. It can be used to perform immunization studies after infection or vaccination. It might be interesting, and this is quite a niche, in order to analyze the efficiency of the vaccination in some specific individuals, for example, in immunocompromised individuals, immunodeficient. And so these are a topic that probably is not related, not developed that much, and it might be very interesting to get to know this information, of course, in order to be able to provide better care for the patient. And also, we can even identify vulnerable patients. So here we are thinking about why not, if we perform this type of analysis to some, again, immunocompromised patients, or even patients with cancer, we might know whether they are at risk or not, depending on the status of their immunization and also their immune status. So yeah, here you have all this information, also the references available for you to take a look at the different papers that we are talking about here. And again, well, the information about both hemat-oncology panels and clinical immunology panels. Okay, so yeah, don't hesitate to write to us at support@cytognos.com if you have any comment or question. And now we will be moving in 15 minutes to the multidimensional and spectral analysis with Infinicyt. This is a talk that we have yesterday at around the same time with my colleague Maria, and we will be exploring how we can perform analysis of spectral files. That you know, these are spectral files from the spectral flow cytometers. These are different technology to the usual flow cytometers that we are using in the clinical flow cytometry, and they require a mixing of the files of the different fluorescent channels before performing the real analysis, and then we can do that multidimensional analysis with Infinicyt. And then, very important, at 5 minutes past 12, will be in this other room, the ESCCA Industrial Panel presentation number 3. This is a plenary session that will be talking about the state-of-the-art reporting for measurable residual disease assessment. So this will be talking about multiple myeloma analysis, and especially focused on how we report multiple myeloma data, and how in Infinicyt and well, the EuroFlow and Infinicyt, and we have developed a consensus report following the consensus scientific information available and references, so that we can standardize how minimal residual disease is reported from flow cytometrists to clinicians in different centers, and through that means we will be able to, of course, obtain more reproducible information, make it compatible between different centers. At the end, we can further develop the kind of research that we are doing by combining all this information together. So yeah, my colleague Maria will be in charge of that from 12 to 12:30, and then we will be back at the lab in Salamanca with our daily session, our daily online demonstration of Omnicyt. Okay, so from 12:30, once the talk is finished, we will be there from 12:30 to 2 p.m. so that you can know more about Omnicyt and its features, its technology, and so on. If you have any comment or question, please don't hesitate to write down in chat. Speak with my colleagues in private rooms, and we'll also be available in the virtual stand. And for the moment, we'll leave it here. We'll stop for a second now, and we'll be back in a few minutes with the session of Maria. I will leave it here. Okay. Okay, so oh no, sorry, it's not correct. We are back at 11:30 for the session. So in 9 minutes we'll be there for this session. Okay, see you in a moment. We are available if you have any questions, write down in the chat. Okay, hi everyone, we're back. So now we are gonna have the second session of the day, well, the third session now. That will be multidimensional and spectral analysis with Infinicyt. This is the same session that we saw yesterday regarding Infinicyt analysis of spectral files and multidimensional analysis with different tools, different parameters, APS, and so on. So I hope that you like it. This will be around 20 to 25 minutes. And then at 12, we'll be moving on to the plenary session regarding multiple myeloma reporting, okay? If you have any comment or question, please don't hesitate to write down in the chat. We'll be glad to help. And my colleagues are also available in the virtual stand and private chats if you have any question. Okay, so you will be watching my screen just now, and you should be listening to the audio.
Hello everybody, welcome to this session called Multidimensional and Spectral Analysis with Infinicyt. My name is Maria Ochoa González and I'm Product Specialist from Cytognos in the Benelux. For the following half an hour, I would like to show you how you can approach the analysis of files coming from the spectral cytometers by using Infinicyt. So first, I would like to introduce you Infinicyt analysis software. Infinicyt is an analysis software of multicolor flow cytometry data, which is compatible with all the type of cytometers and manufacturers. So it's totally compatible with files coming, for example, from the Aurora cytometer. This software has some multivariate tools like multiparametric diagrams and some other tools that we will see during the demo. It is very efficient and sensitive, which is important when processing large files like the ones coming from the spectral cytometers which have a lot of parameters and also a lot of events. Also within the software you have some tools that will allow you to make this analysis reproducible, like the analysis strategy tool and the automated analysis tools. Before starting, I would also like to explain some of the characteristics that are specific from the files coming from the spectral analyzers. We are working with files with a lot of parameters, more than 20, and this kind of analysis is quite complex, so you need to approach it in a different way. Also, in the case of the files coming from the spectral flow cytometers, you need to perform a spectral unmixing on the acquisition software before being able to open the file on Infinicyt. So you need to unmix the file on the software, export the FCS file, and this file you will be able to analyze it with Infinicyt. So now let's see the Infinicyt analysis workflow. So the first step would be to define the main populations that you would like to analyze. Once you define them, you will need to create a desktop for analyzing each population group. And this desktop will include the diagrams with the parameters that you require to identify the study populations. The diagrams can be a combination of dot plots, APS, parameter bands. We will see later an example of analysis and desktops. Afterwards, you will start the analysis by combining the hand gating with the clustering. Once you finish the analysis, you will perform the data interpretation. And in the case that you need to perform a statistical analysis, then you could export the data in a CSV format. So this is an example of the desktop that I have created for this demo. So afterwards, when we analyze the demo file, we will work with a data cleaning desktop. That will be the first step. Then once we clean the populations, the events, we will start splitting them into the main cell lines, so monocytes, lymphocytes, basophils, and dendritic cells. And then we will focus in each of the populations. So for example, we will create a desktop for analyzing all the lymphocyte subpopulations. Then if we want to focus in any of the lymphocyte subpopulations, we will create another desktop, and so on. So you start creating a desktop for cleaning, then you will go one step further and split in main populations, and from there you will go into the subpopulations. In order to perform the analysis, you have a lot of possibilities within Infinicyt. You have, for example, the conventional dot plots, and you can use different representations, for example, the median value. You can represent the contour lines. You have 3D plots. You have the conventional histograms. You also have the box plots. We have the APS. We will explain later during the demo more into detail what is this diagram. We have the population band histogram. So here we are comparing one diagram that we have here in the x-axis within the different populations that we have in each band. And we also have the parameter band dot plots. So here each band is a different parameter, so you can get an overview of the whole phenotype. Once you finish the analysis, if you need to perform a statistical analysis, as I said, you will export the data in a CSV format and make the analysis in a statistical software. Also within Infinicyt, you could create functions and also virtual parameters. So now let's jump into the demo. Before opening the software, I would like to explain you a little bit about the panel that I'm going to analyze. It is a 40-color panel coming from this publication. And the panel has markers that allow you to identify the main cell subsets that you can find in human peripheral blood. Like the lymphocytes, basophils, dendritic cells, and monocytes. So now let's see on Infinicyt how we will approach the analysis. So now let's open the software. For the ones of you that are not familiar with Infinicyt, this is the main window. So in order to open the file, we just click on Analysis, we select the file, and now the file is opening. The first thing that we need to do is to load the profile, that is the analysis template. In my case, I have already loaded the demo ESCCA profile. And in the profile, you always have on the right side the population tree, and on the left side of the screen you have the different diagrams that you use for the analysis. As I explained during the presentation, in this case we will approach the analysis by using different desktop, and with each desktop we will analyze a different population. The first desktop that we need is the cleaning desktop, because first we are going to remove all the debris and the dead cells. So I need to load it, and this desktop contains different dot plots with the markers that I need for cleaning my events. So time against forward scatter area against forward scatter haze for removing the doublets, forward scatter area against forward scatter side scatter, and CD45 against the live death marker. Also, we have here several APS diagrams. APS stands for Automatic Population Separator, and it is a diagram that is exclusive from Infinicyt. So what is doing the APS is using all the parameters that you have in the panel in order to give you the vessel cluster separation, or the parameters that you are using or selecting. In this case, I have just selected the 5 parameters that I need for cleaning my cells. So the advantages of the APS is, one, that is giving you the vessel cluster separation, and second, that is representing all the parameters in 2 dimensions. So in that way, we can get them. So let's start the analysis. We will use a combination of the dot plots and the APS. So first, let's select my cells on the time against forward scatter area diagram. We just select the singlets. Okay, live cells. I remove the rest of the events of the analysis. And now I can continue by using the APS. Here I can get the clusters and then check into the dot plots what is this cluster. So for example, I get this cluster here, and then I see on the dot plots that these cells are dead cells because they are positive for the live death marker. Again, I remove them, and you see that the APS is always recalculating. So the objective in this first step is to clean the file as fast as much as possible, and you know that it's cleaned when you have more or less a homogeneous population. So this is also debris. Let's see. Okay. Okay, so let's leave it like this. Probably we can remove some events more, but for now, for the demo, we can stop here. So once you have your live cells selected, you will start splitting them in the main cell population, so monocytes, lymphocytes, basophils, and dendritic cells. For this aim, we need to switch the desktop into the cell lines desktop. And here I have again some dot plots with the markers that I need to identify the main cell population, so CD45, CD14, HLA-DR, CD123, forward scatter and side scatter. I have some APS with the markers that I need to identify the main cell lines, and in this case I have a parameter band dot plot with these markers. The parameter band dot plot is useful for identifying the cells when I get on the APS, and also for comparing the phenotype within the different populations. So let's start gating. For example, I will gate this cluster here. I see that it's in the area of lymphocytes, but I'm also gating the basophils. So I will redefine my gates on the dot plots by gating the basophils that are CD123 positive and HLA-DR negative. Then I gate this cluster. Those are dendritic cells that are double positive for HLA-DR and CD123, and also in the area of lymphocytes. And now I continue with analysis by using the APS. Those are mainly lymphocytes. I will clean a little bit the population. Okay, now it's recalculating again. This is debris. now I have this population here. This is mainly lymphocytes. Okay, now let's see what this is. This is again debris, so I remove it. And then the rest is mainly monocytes. I can clean a bit on this diagram by inverting the gates. And the rest of the events are monocytes. So now I have finished with the main cell populations, and I can show them. I can display them on the plots and also on the parameter band dot plot and compare the phenotype. If we want to go further with analysis, we will just leave visible the population that we want to analyze at that moment, for example, the lymphocytes, and you will switch the desktop. So now I have another desktop for identifying the different lymphocyte subpopulations. And let's see how to do it. So here I have some dot plots with the markers that allow you to identify the different lymphocyte subpopulations like CD45, CD3, TCR gamma delta, 19, CD19, CD20, CD56, then CD4 and CD8 for the for the T cells, and the CD25 and the CD127. Again, I have some APS with the markers that I need to identify the different subpopulations, and I have a parameter band dot plot with those markers. So let's see how to approach the analysis. I will get, for example, this cluster here, and I check into the dot plots which is the phenotype. So I see that those events are CD20 and CD19 positive, so those are my B cells, and I can also redefine them by using the dot plots. Now I get this cluster here, and I check in the parameter band the phenotype. So those events are 56 positive, CD3 negative, CD19 negative. So those are my NK cells. I can also clean them a bit on the plots, dot plots. Okay, NK, and I remove them from the analysis. Now I will identify my TCR gamma delta T cells, and now I can continue with analysis. For example, I can get this cluster here. Those are my CD8 T cells. Here, those are my CD8 and CD4 positive T cells. Those are my CD4 positive T cells. And the rest are the double negative T cells. So now I have identified all the lymphocyte subsets. And for example, if I want to compare the phenotype, I could leave the different T cell subpopulations and have a look into the parameter band dot plots. If we want to continue with analysis of any of the populations, the approach will be the same. We just leave the population visible and we switch the desktop. So now let's analyze the CD4 T cells. And in this case, I will use another approach for the analysis because I will use the clustering tool. The advantage of the clustering tool is that we are performing an objective analysis, and an analysis that is reproducible and independent from the operator. So it does not matter who performs the analysis because the result will be always the same. So before applying the clustering, I will remove the T-Rex from the analysis by gating on the CD25 against CD27 dot plots. And then I will perform the analysis just by using the parameters that are relevant for identifying the CD4 T cell subpopulations, which are the CCR7, CD45RA, CD28, CD27, CXCR5, and CXCR7. So now let's apply the clustering. I'm displaying. The way the software makes the clustering is by calculating the density for each event and then joining the events according to the density. I will not go into detail for explaining the clustering tool, but if anybody wants to know more into detail how this tool works, feel free to contact us and we will be pleased to explain you more. So now I have all the clusters that the software could find within my CD4 T cells. So let's see how we will approach this analysis. First, I will just leave visible the populations with more events. Okay. And now we are going to use the conventional dot plots in order to identify the cluster. So if we start with cluster number 1, we see that it is in the area of naive T cells according to the phenotype of CCR7 and CD45RA. So it is double positive for both markers. So now I will just create the naive population and select those events and assign them to the corresponding population. If we look into the plot, we see that in the same area we also have another clustering with this red cluster. So I just leave the naive CD4 T cells and the cluster number 5 and I check the parameters. Normally, if we had performed a manual gating, we will just identify one naive population, but the software is able to identify 2 clusters. If we look to the APS, we see clearly 2 clusters, and in addition we can check on the APS which markers are different within those clusters. Oh, sorry. So the marker that is more different within those 2 clusters is CXCR3 and then CD45RA. So the APS is not only useful for gating my populations, but it's also really useful for seeing which parameters are more significant for differentiating 2 cell clusters. Also, it is quite useful to use the parameter band histogram in order to compare 2 clusters, because here we can get an overview of the whole phenotype of the cell populations that we are displaying. In this case, when we display the 2 clusters of naive CD4 T cells, we see that the markers that are different are CD45RA, CCR7, then CD95, CD25 and CXCR7 as we saw in the APS. So as said, this diagram is also very useful for getting an overview of the whole phenotype of the markers that we have in the panel. So for the demo, we will stop here because it is impossible to finish the analysis in 30 minutes. But if you are interested in seeing more into detail how we will perform the whole analysis, or you want to have a look into your own files, just let us know. I hope that you got a picture about how to approach the analysis with Infinicyt. So if you want to get more information about our next generation flow solution, feel free to contact us. You can mail us to our support email, and our support team will be pleased to help you further. So thank you very much for sharing with me those 30 minutes. And I hope that you have enjoyed, and I hope that you enjoy the rest of the session. So thank you very much.
Okay. How are you? I hope that you enjoyed this presentation by my colleague Maria. Someone is asking us whether we can tell what's the instrument that was used to generate the data. I'm not sure what was the exact name of the instrument, but I will check just now. Maybe some of my colleagues can assist me there. Karina, Arancha. But I think it was Aurora. Let me check just for a second.
A lot of parameters, and also a lot of matrices, and mixed afterwards when we desktop have already opened the
I'm just checking the video now, just to make sure.
So now let's open the software. For the ones of you that are not familiar with Infinicyt, this is the main. So in order to open the file, we just click on analysis, we select the file, and now the file is open. The first thing that we need to do is to load the profile that is the analysis template. In my case, I have already the population figure in the presentation, different population. The first desktop, where scattered area against where scattered age. Removing the doublet.
I'm asking my colleagues now, but I'm almost sure that is an Aurora instrument. I will confirm and we'll contact you for that. Confirming that. Yeah, and well, in 7 minutes, we'll have the presentation by my colleague Maria regarding the presentation on multiple myeloma MRD. Let me put that now on the screen for you. Okay. So yeah, now we'll be watching this presentation just in a minute, and you can either watch the presentation here in this room. I'm trying now to connect to the keynote lecture so that you can watch it here, or you can go to that room directly. Okay, so if you go to the program here, you see that now, well, the previous session just finished, and this was a session by Monica Brueggemann speaking also about the minimal residual disease as a well, in clinical studies, and then we want to follow this session with our industrial presentation regarding also a state-of-the-art minimal residual disease assessment. So now we'll be doing that. Okay, I think we can enter the talk here. So now we are entering the room. Okay, and this will be starting in 4 minutes. So I will make sure that you can listen to that. And yeah, this session will be basically covering what are the latest developments that we have developed regarding multiple myeloma analysis and reporting. So first, you might know already a little bit about the databases and how the automatic gating works, but basically in Infinicyt you can take a sample of multiple myeloma. You can take those FCS files, you can put them into Infinicyt, and then Infinicyt will process them. It will actually analyze in a multidimensional space all the events, all the cells, and it will compare all these events, the different clusters, and they will be compared to the EuroFlow databases. They will be compared to the EuroFlow databases, and basically the software will give each of the clusters a name. So it will say, okay, these are lymphocytes, these are neutrophils, these are eosinophiles, these are plasma cells. So then, actually, you will see at the end, the software has analyzed usually 90-something% of the sample. So most of the events will be directly classified as normal cell populations or the Brian doublets, and there will be only a few cells, just a little percentage of the cells that is not exactly as expected by the software. So some of the events are not exactly like as they should be, and this is mainly due to technical differences. So due to the technical processing, there might be differences. Here we have the password message regarding inviting you. Don't hesitate to join us. So session, right? So yeah, this session, sorry, after you obtain all these classification of normal events, then you just need to compare to review that those that are not exactly the same as those in the database, what they are. So you can go through the software and you say, okay, these cells, okay, they are lymphocytes, so you assign them to lymphocytes. What about these other cells? Okay, they are eosinophiles, okay, so you assign them to eosinophiles. And only some of them might be abnormal, okay, so you identify those abnormal, and that's all. So that should take, the software automatically processes a sample in around 10 to 20 minutes, depending on the size of the files. I remind you that this is 10 million events. That's a recommendation. So we are talking about a lot of events actually. So this takes around between 7 to 20 minutes, depending on your computer. And then your analysis of the file shouldn't take more than a few minutes. Okay. So once you analyze the file, then we come to the second step, which is reporting. So Infinicyt automatically includes a reporting report that is automatically created with the data of your file, and actually it includes the consensus, information, comments, and clinical clinically relevant information that has been decided by the EuroFlow experts, by some experts at Cytognos. So we are building this standardized report as a way to standardize how minimal residual disease analysis are reported worldwide, right? By the same information. So it's 10 seconds for the session to start. I hope that everything will be fine. Let me know in the chat if there is something, or go to this room directly. Okay. See you later. Bye bye.
Okay. Okay.
Just wait.
Welcome.
Welcome, everyone.
Yes.
So, my name is Karina Cabrita, and I'm here with my colleague Maria Rodriguez, and we are here to present what are the parameters which are important to include in a MRD reporting. Okay, so we prepared a very nice presentation that we hope is useful for everyone. And it's within the theme from the last presentation. So Maria, please present yourself.
Hi, good morning. This is Maria, and I will, well, I'm a product specialist of Cytognos for over 7 years. So, and I'm glad to be here, as Karina said, presenting how to report your data in minimal measurable residual disease, and how you can report this information with the best quality. So thank you so much for joining us today.
Okay, so you can start the presentation now, please.
Welcome. Thank you all for coming. Welcome. Thank you all for coming to the session. I want to thank Paula Fernandez and also all the organizer of this ESCCA for giving Cytognos the opportunity to talk at, and participate in the ESCCA 2021 virtual conference. My name is Maria Rodriguez, and I am International Product Specialist at Cytognos. And today I will be the presenter of the session named State-of-the-Art Reporting for Measurable Residual Disease. So this is the conflict of interest disclosure. So we allow the ESCCA to publish this session in their website. So now let me start this session by mentioning the main challenges of MRD assessment. And then I will show you how we can overcome these challenges with a state-of-the-art report. Our main goal when studying MRD assessment is to detect if there is remaining disease, because we want to treat the patients with a personalized therapy. But despite the clear need of MRD assessment, MRD assays bring different challenges. Technical difficulties could give misleading results. Sensitive and specific standardized methods for MRD determination are not widely available outside of specialized centers. And this can also be a drawback if in the specialized center they do not have access to this diagnosis information. And this lack of standardization means that the expertise of the operator plays a key part in the diagnosis of the patient. So our main aim when treating our patients is to cure them. But as I said, despite clear and novel therapies, patients still relapse. So as an example, children with acute leukemia, 15 to 20% of children with acute leukemia relapse, and implementing pediatric acute leukemia algorithms lead to substantial increase in the survival in adult patients with acute leukemia. Nevertheless, 40 to 50% of adult patients still relapse. So if we compare the disease to an iceberg, that means that we are not seeing a considerable part of the disease, despite the patient being in complete remission. That is why we need more sensitive techniques that allow us to see what's below the surface to detect measurable, or formerly called minimal, residual disease persisting below complete remission. Note the difference between not detectable and truly negative MRD. The term measurable residual disease has been suggested as a replacement of minimal residual disease because the detection threshold for the various diagnosis techniques are a moving target. The red line indicates the detection limit of cytomorphology. Then we have PCR with a sensitivity level of 10 to -3, and next generation flow and next generation sequences with a sensitivity level between 10 to -5. In-house availability and laboratory experience drive the choice between the different techniques at the moment. The second challenge is that the sensitivity of the MRD assays or the depth of response is very important to treat the patients with a personalized therapy. Patients can respond to the same treatment in different ways. There are patients that do not respond to therapy or they do in a suboptimal manner. And patients who achieve complete remission or even undetectable MRD may experience relapse. Thus, MRD follow-up is crucial for the identification of patients at high risk of relapse. In the last years, we have seen a lot of new therapeutic approaches, and this has increased a lot the survival of the patient. For example, multiple myeloma patients they have an overall survival from 2 to 3 years, and now it's up to 10 years in survival rate in multiple myeloma patients. And as you can see in these images, the 5-year survival improved in all age group, but this improvement is more significant in younger patients, in patients younger than 15 years old. And you can see in this graphic how they have almost doubled their survival rate thanks to new therapeutic approaches and new improvement care. Therefore, this highlights the importance of MRD assessment and how this MRD allows clinicians to have a clearer orientation on what clinical decision they should make, which is patient, and provide them with a personalized therapy. In addition, for MRD detection, we should monitor the aberrant population. An extensive list of aberrant immunophenotypes have been described, including asynchronous marker expression. That is the expression of a marker before or later than expected during the maturation. We can also have cross-lineage, or the expression of an antigen that is usually expressed in a different cell lineage. We can have over or under expression, as well as a ectopic expression. That is the expression of a marker in a population in which is not usually expressed. The pathological cells at diagnosis may have multiple aberrant immunophenotypes, and we should follow all of them in order to increase the sensitivity of the MRD assay. However, MRD evaluation might be studied in referral laboratories, and it's not always possible to have access to this diagnosis information. So after therapy, the monophenotypic aberrancies in a diagnosis may be used as a starting point for MRD evaluation. However, immunophenotypic changes in the expression of at least one antigen between the diagnosis and the relapse were observed in acute leukemia, for example. Furthermore, the ability to discriminate leukemia cells from normal cells can be hampered by the treatment. Antigen-targeted therapies, for example, can result in the decreased expression or even the absence of an antigen that can be used for the identification of the neoplastic population. All these changes in the monophenotype can hinder the detection of the neoplastic cells and might lead to false negative results. On that topic, one of the greatest concerns is related to the quality of the sample. The bone marrow aspiration could lead to blood contamination and the estimation of the burden of neoplastic cells. Other issues, such as sample aging, clotting, and degeneration due to improper shipping or storage conditions, might also significantly affect the results. Evidence of compromised specimen quality includes the hemolysis, as well as the presence of apoptotic cells. To make sure of the reliability of the MRD results, cutoffs to determine the positivity and negative of MRD results should be established. In that regard, the International Myeloma Working Group has defined for the assessment of multiple myeloma the 20 cell cutoff for the limit of detection as a minimum to identify the aberrant plasma cell population. And they have also defined that the aberrant population should have at least 50 cells in order to have in order to obtain reproducible enumeration of cell population, and this is the lower limit of quantification. The International Myeloma also recommends the acquisition of 10 million cells to clearly discriminate between the positive and negative MRD assessment, and the importance of acquiring enough number of events is illustrated on these diagrams. So we can see in red the aberrant plasma cells, and in gray we also have the normal plasma cells. So as you can see, if we acquire less events, we are losing sensitivity, and it's more and more difficult to identify the aberrant plasma cells in between all the normal plasma cells, and therefore it's more difficult to identify or to evaluate the MRD assessment. All of these challenges make MRD analysis very difficult to interpret, and experienced operators are needed to correctly evaluate their MRD results. Therefore, challenging case analysis might carry an element of subjectivity. So, to recap, these are the main challenges we can face when evaluating MRD analysis. And how we can overcome these challenges with a state-of-the-art report. So let me use the multiple myeloma automatic report as an example of a state-of-the-art reporting. EuroFlow has designed and validated different reference databases, including one for the monitoring of minimal residual disease in multiple myeloma. So when comparing a new case with the multiple myeloma MRD database, the population identified are automatically compared with the normal reference ranges for both relative distribution and phenotypes. This information gives us an overview of the patient immune status, and the clinician can use this information to give patients a tailored therapy. Populations outside the normal reference ranges are easily identified because they appear in bold, similarly as in hemogram reports. And depending on the alteration and the affected population, we can conclude on the quality of the sample, the bone marrow immune regeneration, as well as the MRD assessment. For the bone marrow immune regeneration, the major pointers are the precursors and the nucleated red cells. For the quality of the sample, the most important indicators are the decrease in the mast cells, nucleated red cells, and precursors. The modulation is assessed using the frequency of the mast cells. So if they, for example, if the sample sent for MRD assessment is significantly modulated, the disease burden might be inaccurately assessed. And this matters because some treatments or other important clinical decisions are based on achievement of undetectable MRD. And the patient care might be compromised if such decisions are based on an accurate assessment of the MRD. In addition, the quality of the processing is automatically evaluated by the software by separating the bridge, doublets, and nucleated cells. A high percentage of the bridge, above 20%, could indicate inappropriate sample quality or processing errors. The MRD assessment is based on the presence or absence of aberrant plasma cells. But in order to know the exact frequency of these aberrant plasma cells, it's mandatory to identify all the nucleated cells present in the sample. The report also includes the limit of detection and the limit of quantification sensitivity levels. And they are calculated for each sample, depending on the number of acquired cells. As I said, the acquisition of 10 million cells demonstrates an effective sensitivity close to 2 × 10⁻⁻⁶, as described by the International Myeloma Working Group for the Assessment of Multiple Myeloma MRD. The report also includes the immunophenotype of the aberrant plasma cells. So after therapy, the monophenotypic aberrances in a diagnosis might be used as a starting point for MRD evaluation. But in this case, the aberrant plasma cells phenotype is obtained from the comparison with the normal plasma cells included in the database. So therefore, in this case, the diagnosis information is not necessary. Finally, the report will include conclusion about the MRD assessment based on the presence or absence of pathological plasma cells. The report will also describe warnings if the sample is immodulated or if the number of events does not allow us to reach the required sensitivity. Conclusions of MRD positivity or MRD negativity can only be stated if the required sensitivity has been reached. And this is why the limit of detection and limit of quantification have to be taken into consideration. This is also particularly critical if a MRD negative result is obtained. If the result is negative for MRD, but not enough cells were analyzed, the decreased sensitivity of the detection should be properly annotated. Nevertheless, bone marrow aspirates are crucial for MRD assessment and should be analyzed even if we can find some small issues. However, deviation from the expected quality of the sample should be carefully annotated in order to avoid misinterpretation in the clinical practice. You can increase the accuracy and precision of our results by relying on databases created by experts. The automatic report will reveal slight numeric and phenotypic alterations that can be missed, and this is greatly helpful with interpretation of complicated cases that are close to the limit of detection. Finally, after we have performed the MRD evaluation, we can electronically sign the analysis and the report before sending the results to the clinicians, and we can assign different roles with a specific roles for each user in the lab. So in this example, we have the technician that can first certify the analysis and then label the report as analyzed. Then we have the pathologist that can sign off the case and label the report as approved, and then issue when he or she is ready to send out the result to the clinicians. All this together can help you meet the quality compliance and standard requirements of your clinical laboratory. The automatic report is also available in different languages, which facilitate the implementation of this state-of-the-art reporting in any laboratory, regardless of your country. So this is how the state-of-the-art report can help you overcome the challenges of MRD evaluation. So to sum up, according to international consensus, the report should include the MRD level, plus the limit of detection and the limit of quantification of the assay, the proportion and number of neoplastic cells and normal cells, the immunophenotypic expression profile of the neoplastic cells if present, as well as the cell viability and the quality of the sample. This state-of-the-art report can be used for other studies, such as monitoring circulating tumor plasma cells, in peripheral blood for the diagnosis of multiple myeloma, or the screening of, for example, of chronic lymphoproliferative disorders, as well as acute leukemia. There is also available a state-of-the-art report for the screening of primary immunodeficiencies. So this is how the state-of-the-art reports can help you overcome the challenges of MRD assessment. And all of these state-of-the-art reports are available with Infinicyt 2.0. Infinicyt 2.0 is part of Cytognos next generation flow solutions, and we offer, in collaboration with EuroFlow, a complete and scientific-based solution to cover all your needs in your clinical laboratory, including the support from our highly specialized technical team. So we hope that you have gained insight on how you can adopt this state-of-the-art report for the assessment of MRD in your lab. And please visit our booth if you want to know more about the state-of-the-art reporting and Cytognos next generation flow solution. Again, I want to thank all the organizers of the ESCCA for giving this opportunity to talk in the virtual conference of 2021.
Thank you so much for coming to our session. I really hope you enjoyed it. As you know, you have on the right side, you have a chat box where you can write all your questions. Fiona already asked if you can use the database with all the flow cytometers. So the databases were built with the files from Canto because it was what was available at that moment. It really also depends now that we are moving forward. The new databases most probably will be compatible with more cytometers. You can use it with Canto, and with Lyric, you will have more check populations to verify, but you can still use it. With Navius, we also tried it. You have more check populations because the difference is the 4-site scatter, not actually the populations. But anyway, you have to verify more events, but it's still less events than 10 million. But anyway, if you're interested in doing that with different files from different cytometers, just contact us and we can explain how to do it.
In that regard, Omnicyt files are also compatible with EuroFlow databases.
Yeah.
You want to?
Yeah. It's just that you just contact us and we can help you. Yeah, Olivier is asking if we tried with Cytec spectral instrument. So we haven't tried it yet, but if it's possible to make it compatible with the EuroFlow settings, so what is important, it doesn't matter the cytometer, is that you can put the populations in the same place in the MFI. So Infinicyt will rescale the data, but if the 4-site scatter algorithm is really different, then you'll have more check populations for neutrophils, monocytes, and others like that. Okay? Can you? Bojan, I don't understand exactly the question.
Mm-hmm.
So you can work with, there's not a limit of events that you can process in Infinicyt. So if you have 10 million events, you are able to see them.
Mm-hmm.
So there's no issue. So the requirement is the computer. Okay. So if you're working with 10 million events, then you should have a really, really good computer.
Mm-hmm.
If this was not the answer to your question, just please rewrite and we will see it. Okay, so Fiona, it's you're asking if the 4-site scatter is the most important parameters to match up. It's not the most important parameters for the databases because you are looking for other. So you're looking for the MFI markers, so I don't know, CD38, for example. But if you want to look at the complete sample, so the complete immunophenotype of a population, then you want to have your neutrophils or monocytes correctly classified.
Mm-hmm.
Even if they are in the check population, it's very easy to classify. So it's important to have less number of check populations, yes, but it's not important to have a correct classification.
Mm-hmm.
Please elaborate on what is stated on a MRD negative report. So if it's an MRD negative report, it will tell you that it was not possible to find an MRD positive population, and what was the LOD that you were evaluating. It will also tell you examples of the debris that you have, which might indicate that you have like a technical issue. And I think that's it. So, and it will tell you also if it's hemodiluted, for example, because this will affect the results. I don't I don't know. We can have a look later also, Leanne, about this. But from my memories, this is what appears.
Yeah.
Okay. So I think there's no more questions. There's just a clarification from our colleague Jesus. Oh, there's more questions now. Jesus just wants to clarify to Bojan that there's no minimum events to run. So there's no limit. So you can decide what you want to run in your cytometer, and Infinicyt can process it. Just to take into consideration that the less events you have, the lower the sensitivity you will have also. There's a question, are you working on a solution to the problem of PET kappa lambda expression in CRY-tube? So we have no information about this. So,
Yes.
and I don't know exactly which tube you are mentioning, so I assume MMRD.
Yeah.
But we have no information about this. So the kappa and lambda are correctly separated. Maybe the shape is not exactly the same because of so it's not like a round population, but the identification is made correctly. Okay.
So also, Mona, if you can also, well, I think I write privately your. We can also contact you later, and so you can give us more information about this topic.
Okay. So, do you have more questions? We still have around 4 minutes that we can use to explain anything that you have doubts about.
Yes.
Anyway, we have a parallel room that's being managed by our colleague Jesus, where you can enter and ask any questions. So we have also a session about EuroFlow panels later today. So if you have any question, you can ask it there also. Okay, we are also available in our booth, and most of you already have our emails, and you can contact us directly regarding this issue, this team, sorry.
So again, as Karina said, thank you so much for joining us today in the session. So we have some couple of minutes left. So if you have any question or you want to review something, please feel free to write them in the chat.
No. So I think we can wrap up this session.
Okay, Bojan has another question.
Do we have 3 tubes for MRD? We have for MMRD and BCP-ALL MRD, we have 2 tubes. Okay. Actually, most of the tubes, they have common markers, so that you can really confirm that, for example, if you have like 50 events or even less than those events, you have the reproducibility of both tubes. So it's like a confirmation. That's the reason why we have 2 tubes to verify the MRD. Okay.
And yes, also our colleague Jesus is writing that we are going to hold a live demo of Omnicyt in our sponsor room. So if you're welcome to join us also during this live demo.
Bojan, we can contact you later also to explain in more detail all the questions that you are having regarding MRD.
Oh.
Okay. Welcome.
Welcome, Bojan.
Okay.
So thank you so much again for joining us today, and I hope you enjoyed our session.
It will finish now in seconds. So
Thank you so much.
Okay, so I hope that you like the presentation by my colleague Maria. You know, we will be going now to the Omnicyt demonstration, but I just wanted to, for those of you who are here, yes, I would like to take the opportunity to show you actually a sample of one of our reports. Okay, so this is one of the Infinicyt multiple myeloma reports, and you can see how here this is the header of the report, so we can include information coming from the FCS file. Either we can type it in, or it could even come from the cytometer, and it could be automatically updated here. Then we have information regarding the actual result of our analysis. So you see here we have the frequency of our sample. This is the frequency, the percentage of every population in the sample. You see that we are working with we are working with very high sensitivity study here. Okay, so we have 0.024% of normal plasma cells, 3% of B cells, and within B cells, 2.9 are mostly B cell precursors, whereas there are some other mature B cells. And you see that this is for other populations. And then on the right side, you have the reference value for the normal healthy donors. So this is what the EuroFlow database gave us. So you can see how we can use this reference in order to compare against our data. And those values that are highlighted in black here in bold, those are the values that are out of the reference, either because they are too low, for example, normal plasma cells are too low here, or eosinophils are too high. So that's why they are highlighted in bold. And actually in this sample, we also found a abnormal plasma cell population. So that's why it's stating the frequency here, 0.022. And then we have what Karina was mentioning, the limit of detection and lower limit of quantification. So basically in a negative report, in an MRD negative report, we would have here a 0, because if we couldn't find any abnormal plasma cells, it might be 0. Then, but it will also give us the limit of detection. So the point would be actually then you have more comments. So you have here the immunophenotype of the abnormal plasma cells. You see this is the immunophenotype of the population that we found, only kappa positive and with other markers. And then you see we have some comments stating 0.045% of plasma cells were detected, and like 50% of them more or less. So because we have this amount of normal and this amount of abnormal, more or less 50% of them express an aberrant phenotype, and then we have information about that phenotype. Okay, so the conclusion: this is bone marrow compatible with positive MRD, and the number of events analyzed is lower than the recommended standard. So this is what Bojan was mentioning. We don't have the number here in this page, but then the point is that this is below the 8 or 10 million standard that is recommended. So that's why this message is here. If the sample was an MRD negative, then we would have a 0 here probably, or very low number below this limit of detection, limit of quantification. And the ideal aspect would be that you actually report this is a sample negative because I couldn't find plasma cells with this sensitivity, and you report the sensitivity, because actually that's what tells the doctor or the clinician, okay, we couldn't find abnormal plasma cells above this level, but they could be more. There could be abnormal plasma cells below that level, and that's important, because actually that's the difference between let's say a diagnosis where you are trying to find a big cluster or a big population compared to others. So yeah, maybe I will come to this. We can come to this later in the afternoon session, just at the end today. In this session, maybe we can talk more about the multiple myeloma MRD other EuroFlow panels and also about us. So now let's go to the lab. You saw Maria there. She will be working with Omnicyt, and also my colleague Arantxa, she will be in charge today. So now I'm giving the ownership of the presentation to them, and they will be showing you about Omnicyt. And yeah, if you have any comment, doubt, don't hesitate to ask, as always, and we'll be glad to help.
Hi, can you hear me? Okay, I think so. Sorry, because, okay, that's all. So, well, thank you, Jesus, for presenting me, and thank you for coming and joining us in this Omnicyt demo. My name is Arantxa. I work as a product specialist here in Cytognos, and today I am going to show you the Omnicyt cytometer. So in the next hour, we will see the main characteristics or the main features of the Omnicyt, and then we will see how to work with the Omnicyt. We will switch on, perform the quality control, and also acquire a sample stem with the LST kit with the lymphoid screening tube, which is the first tube, the screening tube of the chronic lymphoproliferative disorder. So this is what we, this is our agenda for the next hour. And then we have time for any question that you may have. So don't hesitate to contact us to ask any question, or maybe something is not clear. So just, you are more than welcome to ask anything. Okay, so well, that's the introduction, and now we are going just to start with the machine, with the Omnicyt. So you can see this is a benchtop cytometer. Well, the exact sizes are 40 multiplied by 58 multiplied by 43 centimeters, but well, just to have an idea, it's like a centrifuge, more or less, and the weight is 29 kilograms. So this is the cytometer because indeed it also includes the fluidics. Indeed, if we click here, we can open and see the different tanks. So we can see 4 tanks. Here we have the biggest one, let's say, with a capacity of 1.8 liters, and then 2 smaller ones with a capacity of 175 milliliters. And what contain it of its tanks? Well, the first one is the waste container, so it accumulates all the waste generated by the sample or the used reagent. When this container is filled, we have to empty and again fill with a full straight bleed, so until this line, in order to avoid the contamination of the tank. The next container, the next tank, is the focusing or contain the focusing fluid solution. So the focusing fluid transport the sample, the particle, depending on what we are working with, through the flow chamber. Okay, so it is needed to acquire and to transport the sample. It also contains a preservative and a detergent just to minimize the bubble solution. Then we have here the wash container, what contain the wash solution. So in this case, it is a ready-to-use solution, and the intended use of this solution is to remove and eliminate the debris or the dyes that it may generate and that could make any trouble in the fluidics. And then we have the shutdown solution. It is also a ready-to-use solution. And the use of this shutdown solution is to fill the fluidics when we perform the shutdown, we will see, in order to avoid the bubble solution and also to avoid any crystal or any solid when the cytometer is off. Okay, so you can see that, well, in general, we can say that these containers are small, but it's because the consumption of the Omnicyt is very low. I mean, just to take an idea or to shape an idea, if we are acquiring samples all day, the consumption of focusing fluid, which is the most used solution, is about 1.8 liters per day. Okay, of course, it depends on the sample and the number of samples that we are acquiring, but just to have an idea. Another characteristic of Omnicyt, it is not a pressurized cytometer, it is a volumetric system. Okay, and what does it mean? It means that to acquire the sample, we don't need a vacuum system, okay, to close the circuit. The focusing fluid is enough to transport the sample through the flow chamber. Okay, so, and apart from this, the different characteristic that it allow us is that, for example, we can use different tubes, we can use different flow cytometry tubes, including micro tube, and maybe they could have some split or something, but as we don't need a vacuum system, we can always acquire. Another characteristic, the noise. You can see now the Omnicyt is on, but the noise is minimal, okay? So this is another characteristic that is due to the to be a volumetric cytometer. And another thing is that although we are acquiring the sample, we can refill the bottles, okay? But because when we acquire a sample, when we are running a sample, the Omnicyt acquires the sample, and then we can refill these tanks. Okay, and we are talking about refill, how we have to take or to remove this tank and fill or empty them. Okay, so when we are going to remove them, first it is important to remove or unclog the sensor cable. And then the second step is to remove, maybe you are not seeing, but to remove the fluid system. Okay, and then when we have to connect, okay, again, but just the opposite. First connect the fluid line and then the sensor cable, okay, in order to avoid an increased back pressure and the introduction of air into the system. Okay, so we see the fluidics. Now we will jump into the optics of the Omnicyt. The standard configuration of the Omnicyt consists of 3 lasers, the blue, red, and violet laser, and 30 parameters, 11 fluorescent detectors, and 4 one-side scatter. And in order to localize them, okay, we only need to leave the cytometer leaf, and here we have the optics of the cytometer. Okay, so in this case, in the left part we have the violet lasers here, with all the detectors and mirrors. Indeed, we have here the 4 detectors for the violet, and here we have the detectors and the filters, the mirrors of the red. In this case, we have the 3 detectors for the red laser. And in the right part, we have the filters and detectors for the blue laser, where we have the 4 detectors of the blue. Here, we also have some space. What happened? Maybe you are using or you are performing different studies and you need to change any filter or mirror. Okay, here you can put these filters. So all the information about the optics of the Omnicyt you have placed in the same location. And maybe you can also notice that here we have an empty space. Well, there is the option of an upgrade. You can add a yellow laser that will be here, okay? So in case we add this yellow laser, we will achieve a configuration of 16 parameters, 14 fluorescent detectors, and forward and side scatter, okay? So we are going to close Omnicyt. Okay, so I explained you the hardware very briefly. So now we are going to work with the software, okay? So first of all, of course, we have to switch on the instrument, first switch on the Omnicyt, and then we can go to the Omnicyt software. Okay, So now I am going to use my screen. Can you see it? I hope so. Okay. So first of all, when we switch on, then we will need to log in with a valid username and of course a valid password. Click on sign in and we are in. So this is the main menu of the software. So briefly, we have 4 blue buttons in order to get access to the performance test, new experiment, import experiment, and new experiment for template. They are here because they are the options that are most used, but of course, there are other options. In addition, here we have also more information. We have here the performance test status bar. We will see later, but when we are acquiring or when we are running on the Omnicyt, this status bar changes depending on if we are in the beginning of the process or at the end of the process. Then at right, by default, we have the experiment explorer. So here we have all the experiments that we have in order to quickly access to them. And then we have here the ribbon bar, which contain the 3 most frequently used commands that in this case are file, home, and instrument. First of all, when we are going to work with Omnicyt, we will need to perform the startup. Okay, so going to instrument startup, we are going to start this process. So what's happening? Okay, during the startup, anytime that we have to perform something, Omnicyt is going to tell us. So in this case, we are going to lower the tube lifter and click on next. Okay, so now during the startup, Omnicyt software automatically turns on the lasers and the instrument system, initiate the pumps and prims the fluidics lines. So, okay, this is a warning, this is a clock error. Okay, so we are going to click on OK. Okay, we are going to continue. This is working every time that we have here this color is because the Omnicyt is working. Okay, so now what it is happening is that if you remember when we perform the shutdown solution is filling the fluidics in order to avoid these clogs, okay? So in this case, what we are now is removing this shutdown solution just to get ready the cytometer to start acquiring, okay? So every time that the cytometer is like this, because we have here some colors, some lasers, is because the Omnicyt is working. When it finishes to acquire anything on the process is finished, you will see that this is not flashing, that it will be fixed in green or in blue, depending on the work and the status. Okay, so we are going to just click it and just to, if you can see here, we are performing the startup. And here we have the status bar. So now we are performing the 9% of the process. Okay. Okay. Instrument. Okay. I am going to first unclog because it detects a clock. I am going to load a clean and empty tube. Okay. And click on next. Okay, so I am going to, now the unclog process is performing. You can see here we have the 25% of the process is performed. Okay, well, this is something that can happen when we are in the streaming. So, well, just to see that there are problems that happen, but we can solve them. Okay. Now I'm going to lift. Okay. I'm going to cancel it. Okay, I am going, if let me exit. Okay, I am going to go back again, sorry. Here, this is the Omnicyt software. Then, okay, we are going to try again. Okay, we are going to go back. Okay, we are going to try again to perform the startup to initiate and to be able to work with the Omnicyt. Okay, we are going to try again to close the software. We are going to start again the Omnicyt. Okay. So give me some minutes. Okay. Some moment. Let's try. Okay, you can see this is the same. We can go to instrument. Okay. Mira. Now the cytometer is starting. Okay, let's try it to start up. Okay. So we perform just to remove this shutdown solution. We are going to run again the unclock function. Okay. Sorry for this changes. We are going to load. Okay, this is the same as before, and we are going to perform the unclock function. Let's hope that with this we can continue showing you and performing the demo of the Omnicyt. Okay, it's in the middle of the process. Okay, so well, so now instead of showing you this, what I am going to show you in the meanwhile, while we see what is happening with the software, I am going to show you some information about Omnicyt, which is very used in order to understand better this cytometer. Okay, let me yes, I'm going to share the screen of my colleague Maria and then continue explaining some characteristic of the Omnicyt. Okay, I hope that now you can see my screen. Okay, so we will try to show you some characteristic of the Omnicyt. Well, this is not an aleatory presentation. In most cases, the users and people that can be interested in Omnicyt ask for different questions. So here we include all of them. So just to understand the cytometer and just to try to answer all of your questions. Okay. Okay, we have a question. So thank you very much because, well, maybe now I need more time just to show you the demo. But if you have question, of course, you can ask me. So thank you for this question. The question is, hi, could you run deionized water instead of SID in the instrument? No, it is not needed in this case. You have the ready-to-use solutions and you don't need to use them and to use the SID in the instrument. In the case, as we mentioned before, the most used solution is the focusing fluid, and it is recommended to use the focusing fluid to ensure the quality of the sample. Okay, because maybe also the SID can affect the sample. So with the focusing fluid, we will be able, and we at least the sample will be okay. Okay, so thank you very much. The same, if you have any doubt, of course, you can ask in the meanwhile. Okay, so let's see in the some of the characteristics. Well, as I show you this, I could do it. Okay, the standard configuration of the Omnicyt are 3 laser and 13 parameter. Remember that we have 11 fluorescent channels, but then we have also forward and side scatter. If you remember, the standard consists of the violet laser, the red laser, and also the blue laser. In here we have the side scatter, and then we will need to add the forward, of course, in order to see what is the size, let's say, of the cells or the particles. For the violet, we have 4 detectors. For the red, we have 3 detectors, and for the blue, we have also 4 detectors. As you can see, well, here in this image, we also have the yellow laser. This is an option. I am going to point it. Okay. Here we have the yellow laser, and here we have the space to put more filters or more mirrors, depending on the studies that we perform. As we mentioned, well, for the violet laser, the laser emit at 405 nanometers, and these are the different options that we have. In the blue laser, these are the detectors. Also, the blue laser emit at 488, and these are the detectors and filters that we have in order to get the information. And finally, we have the red laser detectors that emit at 635. And here, well, using the different band pass filters or short pass, long pass filter, we can detect in this signal into 3 different detectors. How we can exchange or how we can put new filters in case that we need it, okay? So it is very easy because these filters and mirrors are very easy to change. So in those cases, we just need to get them, to extract them. They are very, very small. It's like a finger, let's say, of the longitude of these filters is very, very small. So we can change depending on our needs. Okay. Another important thing, and a lot of people ask for, is the flat top laser, because this is another characteristic of the Omnicyt. The lasers has this type of, or we have this type of laser, the flat top. That means that they are aligned, okay? So if we compare, I think that maybe in these images you can clearly see what are the difference between a flat top laser that Omnicyt has and the Gaussian laser that other cytometers has. So in this case, we can see this is the flat top, okay? This is not, this is different comparing with the Gaussian. The Gaussian is like a point, only a dot, let's say. And then the flat top are flat, okay? So they are aligned. So you are going to excite with the most exciting part of the laser to more cells. I mean, in this case, we can clearly see if the cells pass through the center of the laser, okay, they are going to be excited with the more excitation part of the laser. But if the cells are not going to the center of the laser, maybe from one side, as it is a flat top, we can assure that we always are going to hit them with the more exciting part of the laser. What happened with a Gaussian? Okay, so in this case, maybe on the one when it is correctly aligned, okay, we are going to hit the sample with the more important or the more exciting part of the laser, okay? But day after day, okay, the laser, the alignment is not as precise, maybe it can change. So in these cases, when we hit the sample or the particle, depending what we are working with, maybe we are not going to hit the cell with the more excitation part of the laser, with the most intensity of the laser. So the information that we want, that we obtain, it's not going to be the most accurate. Okay, so these are the characteristics that I would like to show you. And how it help us? Well, if we use flat top laser, we are going to minimize the effects caused by the instability and alignment issues. As we see before, although we could have an disalignment, we are going to hit with the most bright or the most intense part of the laser if we compare with another type of lasers. So this allows us that the calibration will be less frequent due to this high stability and sensibility of the instrument. Okay, this is another example showing us the flat top laser. So here we can see, as in the previous slide, on the one we are hitting with the most bright part, with the most intense part. In the day 45, we are also hitting with this part, which does not happen with the Gaussian laser beam. Okay. And what is the contribution of this? Okay, so the instability, we don't need to calibrate so often as with other cytometers. Indeed, here we have the standard deviation, so you can see that here we have 5% up or down. Okay, so in 45 days, in 146 days, the rainbow beads are within the 5% up or down, taking into account the middle, the reference. And then if we compare with any cytometer that have a Gaussian laser, we will see, or we can see, that the changes are very, very clear. And although this is the time period for this is shorter. Okay, so now, well, while I was explaining you these options, Maria helped me with the cytometer. Okay, so just to be able to continue working with it, so I am going to start sharing my screen. Okay, I am going to make host my computer, the computer of the Omnicyt, so I can continue showing you the cytometer. Okay, let me know if now you can hear me, because my colleague said that. Okay, thank you. Okay, so we are going to continue. As we mentioned, we finished, my colleague Maria switch off and switch on the Omnicyt and perform the startup function because it was ready to perform it. So now we can continue with the process. Indeed, as I mentioned, we obtain a green color here. So that means that it is ready, so we can continue working with it. Okay, so before acquiring the sample, we will need to monitor the daily performance of the Omnicyt. So indeed, this warning here is telling us that we have not performed the quality control today in the last 24 hours. So to perform this quality control, we have the performance test. So the performance tracking is the procedure that allows to ensure the accuracy and the sensitivity of the cytometer, and it provides also, we will see, information about the lasers and detector channels. So we are going to run this QC, this performance tracking, selecting the performance test icon. Okay, here in the main menu. And now you can see that the screen changed. Now we have here all the steps that we have to follow to get ready the instrument, to perform the quality control of the instrument. So first, we have to check the filter to verify the instrument configuration. So we can click here. And now we have here the information about the filters that we have already seen at the beginning and also in the presentation, okay? So as it is okay, we can close it. What is the second step? We have to verify the bit lot number, because for this performance test, we need to use the performance test bits or the PTBs, okay? So we need to verify that the lot that we are using is the same lot that we have here. In the case that we have to change it, we can do it, okay? And then we have to prepare the bead suspension to perform this quality control. So as you can see here, we have all the information. We have to add 3 drops of performance tracking beads in 2 milliliters of focusing fluid. Okay, we need to mix very well the performance tracking beads just to homogeneize, just to shake it very well, and then perform the solution. And then we will need to acquire. Okay, so now, well, my colleague Maria prepared these performance tracking bits, so we are going to acquire them. Okay, so they are here. I think that you can see them. Okay, so mix well, and then we can put it in the tube lifter and up. Okay, load the tube. So that then the next step is to run performance test. So we can click here. Okay, you can see the cytometer is working. So here we can see that the laser flash because the cytometer is working, but also we can see that the screen changed. Okay, as you can see, when we click on run, the software automatically shows us a workspace. We don't need to create it. This is automatically created with 4 dot plots where we can see the forward side scatter. In the forward side scatter, we can see the 2 populations that are contained in the performance tracking bits. And then we have 3 more dot plot, each of them corresponding to the blue, red, and violet laser. Because these fluorescent, these performance tracking bits emit for all the fluorescents. Okay, so then in each one, we can see 4 different intensities. This is because each performance tracking bits contain a mixture of bits of flow fluorescent emission intensities. Okay, so the emission intensities vary from the negative to the more bright signal in order to ensure the accuracy and the sensitivity. And also, these performance tracking bits will be acquired at all the flow rates that, if you remember, or if not, I am going to say these are the 12.5, 25, 100, 200, 500, and 1000 microliter per minute. Okay, in addition to that, we also have here the progress information about the progress of the process. So you can see that the process already finished. So this is the information that we obtain when the process is finished. As you can see, the process takes more or less 5 minutes, let's say, to complete, and this is the required time for the laser to warm when switching on the instrument. So when we finish the performance test, the instrument will be ready to be used. Okay, so in this case, the table with all the information that we obtain, just to mention the coefficient of variation as we see in the presentation. This here we can see the variation of each of the fluorescents in comparison with the references. So if we compare with the references, we can see that the coefficient of variation is less or is lower than the 5%. If you are familiar with EuroFlow, EuroFlow accepts a variability of 50%. So, okay, we are in this accept range, okay? And this is the information that we obtain for today, but there is also another option to obtain the information. Indeed, if we go to a Levegenin report here, we can see the variation of every of the forward side scatter, but also for the different lasers. Okay, so here we can see the variation that it could be well, in this case it could be big, but if you take a look, they are only in the biggest case, let's say 4 voltage of differences. Okay, if we go to the blue, we'll say the same. The changes or the voltages that we change are not so big. Okay, the most cases the biggest one is 4, and here maybe we have one of 6. Well, you can see here in some cases, okay, we have a red dot. Don't worry about it. This is because we don't shake well the performance tracking bits, so we cannot obtain the statistics. So that's why all this information is in red. So that's why it's also important to check very well the performance tracking bits. So this is the 15th of December. So if we click here, we can see that, okay, in the next test that we perform, when we check correctly the performance tracking bits, we see that all of them were in green because all of them are okay. Okay, so well, you can see that I am going to back to the current PD results. So you can see that the results are very robust, and this is a consequence of the flake top lasers as we see before. Okay, so Omnicyt use these flake top lasers with an intensity profile that allow a much wider window of alignment. So the use of this type of lasers guarantee the most reproducible and accurate fluorescence measurement possible, and at the same time, it reduces also the frequency of system calibration. Okay, so now we can see also in the right part that everything is okay. So we obtain a ready green icon, so we can continue with working with the Omnicyt. So let me go back to the main menu to start acquiring the sample. So if we are following EuroFlow protocols, once we check, once we check and the instrument, we check that the lasers work properly, the instrument is ready, we need to calibrate the cytometer. So the calibration process allows us to obtain the events within the plot, but on top of that, we will be able to compare the results obtained from different cytometers and different centers, since all the cytometers will be equally calibrated, so the results that we obtain will be comparable. So for that, according to EuroFlow, we need to set up the forward side scatter and also the fluorescent. So for the forward side scatter, to set up the forward side scatter, we perform this experiment. Okay, I am going to double click just to open this experiment. Okay, you can see that the workspace changed. So in this case, we perform this experiment with lyse peripheral blood, just lyse and wash, and then acquire it. If you have any doubt about the methodology and EuroFlow protocol, just contact us and we can go also in deeper. But in this case, we acquire this type of samples. Okay, here we have the image that we obtain after acquiring, and then we get here in the lymphocyte area. Then we adjust the intensities for the forward side scatter for the recommended target values. You can see here, for the forward, we adjust to 220,000, and for the side scatter, 52,000. Okay, if you are familiar with EuroFlow and maybe you have a Canto, okay, you can see that this is 4 times the target for Canto, because the resolution, the numbers of channels that Omnicyt has is 4 times the Canto channels. Okay, so that's the reason of these values. Okay, so in this case, allowing and getting this information and the lymphocyte in this area, we will be able to always obtain the lymphocyte in the same place. In addition to the forward side scatter, as we said, we will need also to check the fluorescent. For that, let me open this experiment. We use the rainbow beads. So here we can see different plots showing the bits in the forward side scatter. So here we identify the bits, and then in the FITC versus PE area, we adjust or we select the 5th peak. For the Omnicyt, it's important to select the 5th peak. Then we have different histogram for each of the fluorescent with the corresponding statistics here in the right part. Okay, so as in the previous case, they are defined or are defined some target values that we need to read in order to be able to always obtain the fluorescence and the population within the dot plot. Okay, in this case, if you need this information, you can obtain the target values for this fifth peak in our website. Okay, so in this case, with they are within the range, so all the events will be placed within the diagram and in the same position. And of course, as we mentioned before, the result will be comparable. However, I am talking about EuroFlow, and if you are not working or you are not following EuroFlow methodology, don't worry, because if you follow any other standard or panels, Omnicyt is also for you. Okay, so once we check and confirm that the forward side scatter and the fluorescence are ready, we can run the sample. So now we are going to create a new experiment for the compensation controls of the LST tube, which is the tube designed for the screening of chronic lymphoproliferative disorders. So for that, we can right-click over the username, that in this case is demo, and then click on experiment. This is one option to access or to create a new experiment, but you can also go to file and new experiment. And then the new experiment dialog open, where we have to name the experiment, for example, LST compensation. Okay, and then we can choose between the different groups. Of course, if we are working with a tuba plate, depending. Okay, in this case, we are going to take as an example one. So we are going to create it. Okay, so after clicking on OK, the desktop, you can see that it changed. Okay, that where we can see different options. So at the right, again, we have the experiment explorer with all the experiment that we have, but also we have the instrument setting tab to access to all the settings. Okay, so we have information about the different parameters. If we click here, we have here all the different parameters. Okay, in this case, as we are working with LST, we need only 8 parameters that are contained in the LST. Again, we can choose between area, height, and width, depending on the information that we need. Then we have also the voltages settings, just to adjust the voltage for each of the channel, including, of course, for one and side scatter. And then we have the threshold. Okay, so here we can set the minimum signal level to eliminate the unwanted events and reduce noise. So that means that all the events that are below this parameter, this value, are going to or are considered debris. Okay, in addition, here under the threshold, we have the exclude coincident event checkbox that when it is checked, it allows to remove the coincident event from the data. So depending on the studies that you perform, you can consider to check or uncheck. By default, it's unchecked. Okay, so once we explain the instrument settings, what we are going to do is use and apply the voltages that we have defined for each fluorescent channel in the rainbow experiment and apply to this current LST compensation experiment. So for this, let me go back to Experiment Explorer, select the instrument setting of the rainbow beads experiment, and drag and drop to my current experiment. Okay, so now if we click OK and go back to the Experiment Explorer, we can see, if you remember, here we have 40. Now we have 10 because we drag and drop the information of the previous experiment. Okay, so we can change it, but then all the voltages are the voltages, all the voltages that we need to acquire the sample. Okay, so now let me go back again to the experiment Okay, so now what we have to do as we are performing the compensation is to name the target of each of these channels. Okay, so instead of put FITC, what we can do, as it is, we are going to acquire a generic, FITC is generic, a generic fluorogram, we are going to put here generic. For the P, we are going to do the same. The P is a generic fluorogram, so we are going to put generic. In the case of the PCP, a 5.5, this is a tandem dye, so in this case, we are going to name with CD5. Okay, and then here for the P cyanine 7, again, this is a tandem, this is a specific, it needs a specific compensation, so we are going to put CD19 this year gamma delta. Okay, if we continue here, we don't have checked the second channel of the red laser and the third and the fourth of the violet because they are not included in the EuroFlow standard configuration, and they are not needed also for the LST. Okay, in this case, the APC is also a generic fluorogram, so we can put it, and in the case of the APC, it is unspecific because it is a tandem dye, so we can put CD38 for the plasma cells. For the Pacific Blue, it is a generic, and finally we have OC, Orange Cytognos 550. So in this case, similar. This is a generic fluorogram, but not all labs. This is our antibody. So in this case, we always include in the tube. So you can also put this information here, although it is a generic. Okay. So once we finish renaming the antibodies, I am going to go back to the Experiment Explorer to create the compensation controls. So for that, we just need to right-click over the compensation and select compensation setup. Then you can see that the setup, compensation setup dialog open. So here we have to choose between different options. So first of all, the source, in this case, they are tubes, the measurement, depending if you want to measure the compensation using the area or the height. And then in this case, the area, and then we have the option of select the negative control. In this case, we can use the negative gate. Just to mention, these compensation controls were created using bits that contain a positive and negative population in the same sample. So we are going to use the negative gate, but imagine that you are working with peripheral blood and you have different peripheral blood stained with different fluorogram, and then you have an unstained control. So in this case, you can select this option. And then you have here also the parameters, the compensation parameters. So if you see, they are the same parameters that we select in the instrument setting panel. Okay, now we can click OK. So now this dialog box closes, and here it creates or updates the compensation control for each selected parameter in the Experiment Explorer. Moreover, the compensation workspace open automatically, so you don't need to create it, with all the necessary plots and gates to perform the process. So here you have a dot plot of forward versus side scatter, just to select between bits or plots, I mean, just to select the reference population. And then on the right, we have a histogram to select the positive in green and the negative population. And this is something that we have for all the compensation controls. Okay, so now we can start acquiring the sample. In the left, we have the collection panel, which contains controls and contain the indicators used for the acquisition of this type of samples. So we have here run, record, recover, we will see, but also we have different flow options. Indeed, we have the flow, the acquisition volume we want to acquire, because if you remember, Omnicyt is a volumetric system, so we can, or we need to put what is the volume that we want to acquire. In this case, we are going to put 50 microliters, okay, just for these compensation controls. And then just below, we have the information about the flow rate. The flow rate of the Omnicyt varies from you can see here, you can move it here, from the 12.5 microliters per minute, 25, 10, 200, 500, and 1000 microliters per minute. Okay. In addition, a special feature of this instrument is that the acoustic focusing option is applied from the flow rate of 100 microliters per minute. So the acoustic focusing at least allows to align the particles or the cells into a single line, into a focused line along the flow chamber, independent of the fluid flow. So in other words, the particle will be passed one by one through the excitation lasers in order to obtain more robust results. And it is very important or useful when we are acquiring, imagine minimal residual disease sample, when you need to acquire large volumes of events, 10 million of events. So with Omnicyt, you can acquire samples at high flow rate as 100 microliters per minute without compromising the quality of the acquisition, because you are not going to acquire doublets, since the cells they are going to pass one by one through the interrogation point. So you are going also to save time. Okay, so therefore the acoustic coupled with the flat top laser results in more robust and reproducible data. But in this case, for the rainbow beads, we are going to put in 25 microliters per minute. And then we also have the stop options. I mean, we can select the event we want to acquire before stop. In this case, we are going to put 5,000, okay? And well, this is all that we are going to select for the collection panel. So once we select all the desired option, we can apply the same acquisition condition to other samples, just selecting to apply to experiment options, okay? So with this, all the compensation control will be the same, will have the same stop conditions. Okay, so now we are ready. We are ready to acquire these compensation controls. And for that, we are going to start with the first channel of the blue, which is the FITC. So for that, you can see here, my colleague Maria take the FITC, give me the FITC. So I am going to remove the PTB tube and put FITC, okay? I can put it, okay, up, and then we can press run. What we can do is wait a couple of seconds just to see how is the acquisition of this event. We can see we have here. So now we can record this information. So now you can see very clear that here we have the beats. What we can do is just to adjust better. If you don't have, if you are not very good at it, okay, just to simplify, you can zoom in, and then of course you can move and you can modify the gate just to adjust better to your sample, okay? So something like this. I am going to put this also here, so you have to adjust better, and then just to zoom out. Okay, so we already acquired this FITC compensation control. Of course, we have here the negative population, but we can adjust a little bit if you want, and the same for the positive population. Okay, so we finish with this FITC. Okay. What happened? Okay, let's see in the collection panel. We only use, if you remember, we put 50 microliters, but we only use 17 microliters. So we can obtain this the remaining sample, because As Omnicyt is a volumetric system, once we acquire the design number of events, we can recover the unused sample. So for that, we can press on recover, and the remaining sample is returned into a clean or in the same tube as in this case. Okay, so we can click on next, and then this unused sample will be go back to the FITC compensation control. And this is because Omnicyt has a loop before the flow chamber. So these loops detects if there is any bubble or clog before the sample. So if it detects any clog, it will stop the acquisition. But it also, as in this case, it allows us to recover the unused sample. Okay, so I lower the tube lifter. I am going to continue with the next compensation control. We can click here, or we can also select here the second one. There are different options to change. Okay, so now we are going to continue with the PE. If you remember, it is a generic compensation control. Okay, so now we can press on run. Okay, so that's the same. We can wait a couple of seconds, and then what we can do is press on record. Okay. So now here we have the information about the collection panel. So now we can see the remaining volume, okay, of the sample that is being acquired. We have also here the percentage of volume that is already processed and the number of events that are being collected. Okay, so we can see here that, well, we don't have so much positive population for PE as for FITC. Okay, so we can do the same. We can adjust just using this gate. Okay, as in the previous case, we can zoom in and then adjust it. Okay, zoom out, and here we can select between the positive population, okay, and the negative and the positive population. Okay, just to assign better here, okay, and we have you can see here the status bar, how we are. We perform the 70% of the acquisition of these compensation controls. Here we have the information about the events per second of this sample. Okay, so now if we wait a couple of seconds, they will be finished. Okay, here also this is the same information, the status of this acquisition that is almost finished. Okay, let's wait a couple of seconds. Okay, in this case, what happened? If you can see here, we only collect 3,000 events. So what we can do is to append, add more events to this population, to this record. Okay, so for that, we are going to click again on run, but before that, I am going to, okay, so we are going to click okay, so we can lift the tube lifter, we can take it, okay, and then we can press again to run. So if we press again overrun, okay, we have to wait a couple of minutes of seconds and then press again on record. So when we press on record, we have these two options: to overwrite in case that you want to record this new data, or to append. Well, this is what we are going to do in order to achieve the desired event. Okay, so now this information is adding to the previous information. Okay, so in this manner we will achieve to the desired number of events. And you can see also that in this case, the speed is higher, okay? We acquire all this information very quickly. Okay, let me recover the sample. Okay, we are going to click on next. So we are going to recover this sample in the same tube. Okay, I am going to lower the tube lifter. Okay. First, or before go to the next compensation control, what we can do is apply this gate to all the controls. So we don't need to change or to modify this gate in all the cases, okay? So click this one, and then we can change and we can go to the third compensation control that, if you remember, is the PCB cyanine 5.5. And then click on run. Okay, so you will see the information out. Now the software is acquiring the sample. Okay, you can see here we have the gate for these bits. Okay, here we also have the information about the positive and the negative population for this PCB cyanine 5.5. Okay, so what we can do is modify this gate in order to adjust better to the negative area, but also to the positive area. Don't worry, I forgot, let's say, for one moment, we can forget to press the record button. In this case, I do it just to show you that we also have the option of autosave. So in any case that maybe you forget to press the record button, there you will have the option to save also this information, because the Omnicyt stores this information as a backup, let's say. So then we can recover this information without any problem. So don't worry if you miss to press on record. Okay, so you can see here, of course, we are not recording, so this status bar is not loading, okay, but we can see here what is the remaining sample. When the remaining sample is finished, okay, you will see that the acquisition of this compensation control will be finished. Okay, so this is just to show you another option that we can perform on Omnicyt. Okay, so let's wait a couple of seconds. It's almost finished. Okay, so you can see it's finished. Now we have the option of save. Okay, so if we click here, this information automatically updates, and now we have the information already loaded and saved it, sorry, in the to the corresponding compensation control. Okay, so let's do the same with the fourth one. Okay, the fourth one is the PCyanine 7. It's also a tandem die, so it needs a generic compensation. Okay, press on run. And then when we have the record option available, and when we see that the event and the sample is okay, we can press on record. And we can, of course, adjust this. Well, the objective of the compensation, well, at least the compensation is the process to correct the fluorescent spillover. So to remove the signal of fluorescence from all detectors except the one that it belongs to. So that's why we need to also perform the compensation. And we have two types of fluorochromes, and that we see before. We have the generic fluorochrome. So these fluorochromes have the same emission characteristic no matter what conjugate or provider. We are going to remove the PCyanine 7. Continue with the APC. For example, the APC is a generic fluorochrome. So no matter the lot or the conjugate or manufacturer, the the emission characteristic will be the same. Click on here, run. Okay, but there are other fluorochromes that need to be specifically compensated. Okay, let me record it, because the spectral characteristic can differ from conjugate to conjugate, from lot to lot. Okay, so we need this specific compensation. Okay, here we can also select the negative area, and the same for the positive. Okay, so that's our APC, which is generic. Okay, we can see again the remaining, the sample that it is not used. And of course, we also have here the information about the event that we record. Okay, so we have to wait. Here we have the, well, we almost complete this process, okay? So let's wait some seconds. Okay. So let's continue. Next one. The next one is the APC C750. We are going to, is in R3, run, and then we will need to, when this option is available, to record this information. Okay, in this case, the APC C750 is conjugated with the CD38, which is used to identify the plasma cells. Of course, we can adjust better the positive and the negative population. We don't need to wait until the process finishes. We can adjust this population while we are acquiring, but of course, you can also wait until this process is finished. Okay, so we are in the middle of the process. Okay, here we have also this information that we mentioned, and here we can see that we finished with the rail laser. Now we are going to start calculating the compensation of the violet laser. Okay, so for the first one, if you remember, this is a generic fluorochrome, the Pacific blue. Okay, so let's click on run. Okay, so the software, the cytometer takes this volume, okay, now pass through the flow chamber, and now we can record this information. The speed or the flow rate, as we mentioned, is 25 microliters per minute, and the stop conditions are 5,000 events. So when we acquire this number of events, the acquisition will stop, although we have maybe some sample that we can recover. Okay, the same as we mentioned, we can do this during the process when it is acquiring, or then to finish, or when it is finished. Okay, and the last compensation control, which is the CD45 OC or Orange Cytognos 550. Click on run, and then the cytometer takes the volume, and now we can record this information. So we are going to do the same. We are going to select the positive population. Okay. Sorry, the negative population and the positive population, the negative in blue and the positive in green. Okay. So we almost finished with the compensation. We almost finished to record the compensation controls. Just need some seconds for it. Okay. Okay, we are almost there. So just we finish with the acquisition of the compensation controls. What we can do is see the compensation matrix, which is automatically calculated. So for that, we can go to compensation and view matrix, and here we have the compensation matrix with the values that will be now calculated. Okay, so until here we have the experiment with the compensation controls of the LST kit. So what we can do is export these compensation values of this kit. So we can export, click or right click over the compensation and export compensation. And here I am going to put this information in the desktop just to have it more available. And here we can see LST compensation controls. Okay, so now we have this information in the desktop. So then if we need to apply this compensation matrix and values for any experiment, we can do it. Okay, so after calculating the compensation matrix, we can acquire and we can create a new experiment for acquiring the sample stain with the LST tube, the first tube for the screening of chronic lymphoproliferative disorders. So for that, as we see before, we can create it just right clicking over the demo, the username, and click on new experiment or using the file. But now I am going to show you how to do it from a template. At least a template is it's a profile that contains a lot of information, all the information that as the stop condition, the acquisition volume, etc. that we will see now. Okay, in this case, I am going to select the LST template, but you could have here the templates depending on the studies that you perform. Okay, so click here and then go to next. Okay, in this case, we are going to name as experiment, as LST, sorry, LST sample, and then click on finish. Okay, so we have this new experiment here. If you remember, we create in this case, well, this is the name, but we can do in order to save time is just to use the barcode reader. So instead of rename it, just writing or whatever, what we can do is if we have a barcode reader, which you only need to have it, you can put the name automatically using this device. Okay, so in this case, well, our patient is John Smith, or we can imagine that is John Smith. So now we have this information available. So using the template that at least contain, if we go to the Experiment Explorer, all the information already available, instead of including the stop conditions and also the acquisition volume, we can also save time using this barcode reader. Okay, so as in the previous case, in this case, if you remember, for the compensation control, we can drag and drop as we previously performed from with the instrument settings, or what we can do is import the compensation that we just create. Okay, open it. Now we have these compensation controls with the previous compensation matrix. So now we will have this sample available to acquire, and then the information or the step that we need to follow will be the same. If we have the sample, we have to put in the tube lifter the sample, press on run, and then we will acquire a sample stain with the LST kit. Okay, so once we finish, what we have to do is go to instrument, and then here we have to perform the shutdown. What we recommend to do is the standard shutdown. In this case, you are going to fill the fluidics of the Omnicyt with this shutdown solution. Okay, so just to avoid the bubbles formation, and we don't need to perform anything. Why? Because the Omnicyt has the simmer mode, so the lasers are working or only when during the acquisition. If they are not acquiring, they are switch on, okay, switch off. Okay, so this is going to save and to prolong the life of our lasers. So when you perform the shutdown solution, the shutdown procedure, you don't need to switch off the instrument. You can leave it and switch off the computer, but leave the Omnicyt. Okay, so now we finish with this Omnicyt demo. I am going to, if you have any question, of course, you can share with us these questions, okay? I am going to share I am going to make host my colleague Jesus so he can present. So he can present the next topic. So thank you very much, and we stay in contact if for anything that you want or you need.
Hello everyone again. Thank you so much, Arantxa, for this nice demo. I hope that you liked it. As always, I would like just to remind you that this session is being recorded, so this will be available in the coming days on our website and also here in the ESCCA material. This will be available as a whole video, and you can just select what session you want to watch at a given point. Okay, so let me share my screen. We'll be having now the next session, your cheat sheet for smarter clinical flow cytometry testing. This is a session regarding tips and tricks on flow cytometry, different steps of the flow cytometry processing, and then we'll be going with the afternoon sessions, Omnicyt again, focused on multiple myeloma MRD and high sensitivity studies, T-cell immunotherapy research related to some panel, a new panel, an innovative panel for the study of T-cell subsets, and this can be used both for research and also clinical trials. And then we'll have the last session where we are available for all your doubts or comments regarding EuroFlow panels and some recent developments on that side. Let me check. I think we have some comment. I have a question about exclude coincident events checkbox. How do you? So we have a question. I will just show here in the text. So I have a question about exclude coincident events checkbox. How do you make sure they are not the events of interest? Maybe my colleagues can assist me there. I'm not sure of what you mean. Probably Arantxa and Maria can assist there, because I think you are referring to some option in the Omnicyt, and I think it is related to the gated strategy in the Omnicyt software and how you select the events. If that's related to that, I would like to say just that the gating strategy doesn't actually change your data, and that's an important thing. So whenever you are analyzing your data, because you have these exclude coincident events, I think it won't. All the info still will be acquire. So the information will be saved in your files, and you will have access to that. They will not be lost. Still, let me check with my colleagues, and we might contact you later, Bojan, in order to clarify that. And now I will pass the leadership of this presentation to my colleague Karina, who will be in charge of the following sessions. Okay, so thank you so much, and see you later.
Okay, so I'll be in charge of the afternoon sessions. Bojan, as we said, so we can arrange a meeting and solve every question. So actually, Maria, I'm sure she's in a break now. So when she comes back, she will answer this question. Okay. So I'm going to share my screen now. Okay. Just check the.
Okay, can you, I think now you can hear me better. Okay. Okay, fine. So I'm going to share the presentation about your cheat sheet for smarter clinical flow cytometry. Okay. And then we have some times that we can.
Welcome everybody.
Okay. So I hope you enjoyed this session.
Welcome everybody. Thank you all for coming to this webinar. My name is Irene, and I'm presenting today your cheat sheet for smarter clinical flow cytometry testing. These are the points that we are going to cover today. First, I will start off highlighting the value of flow cytometry in clinical practice. Then I will talk about the current issues we face in the clinical routine lab. And I will focus on the specific challenges of the minimal residual disease assays and on the design and validation of flow cytometry tests. Finally, we will see how we can overcome these challenges with a fully standardized solution, the EuroFlow method. So, what is the role of flow cytometry immunophenotyping in the clinical lab? It allows the diagnosis and classification of leukemia, lymphoma, and plasma cell dyscrasias, and the detection of minimal residual disease after treatment. Flow cytometry is also used for studying the immune response of patients and for progress control of the treatment itself, and in the clinical immunology field to support primary immunodeficiencies diagnosis. Now, flow cytometry is definitely a versatile and powerful technique with many applications in clinical settings, but it's also complex and it has multiple steps. First, we collect the specimen, transport it to the lab. Once it gets there, we process the sample, we stain it, and acquire the tubes in the instrument, which has been previously calibrated and compensated. And last step is analyzing the data and reporting the results to the clinicians. So there are multiple checkpoints here where we can introduce errors, especially in the pre-analytical and post-analytical phase. And these errors have economical consequences and negative effects in patient care. And that's why all flow cytometry assays require careful assay design, instrument setup, performance validation, and ongoing quality monitoring. Now let's see what are the common challenges we face in clinical routine. First, sample stability can be an issue. Most of times we require a fresh sample, in some cases no longer than 36 hours after collection. The samples that come in are very diverse. We get blood, bone marrow, lymph nodes, and they come with different anticoagulants like EDTA, heparin. So they require different shipping and storage conditions, especially with regards to the temperature. There are different WASLICE protocols in place, so we have to coordinate multiple workflows at the same time. Cocktailing the single antibodies can lead to pipetting errors, and if we get a new lot, we need to cross-check it with the previous one. And we need to have an efficient stock control of all the reagents to reduce reagent waste. Now, our instruments have to be correctly set up, and we need to track the day-to-day performance to ensure consistent data over time. It's important to acquire the tubes as soon as possible to minimize sedimentation and not to compromise the quality of the sample. And during the acquisition in the instrument, we can run into issues such as unstable flow, clogging, or air bubbles. When it comes to the analysis, there is always a user bias because positive and negative populations are clear, but there is a lot of middle ground in between. So the final results highly depend on the analyst expertise. And lastly, we have to put together a report with our lab test results, transfer it to the LIS system so it can be accessible to the pathologist and the clinicians, while ensuring that all the sensitive data is protected. So these are the common points regardless of the type of study. However, more sophisticated tests, such as minimal residual disease assays, pose additional challenges in the lab. To follow up patients, we usually analyze bone marrow, which is a very delicate specimen. Sometimes we only get a small volume to run the test, and we can have hemodilution issues if we don't get the first pool. Plus, the anticoagulant may affect marker expression. For example, heparin downregulates the CD138 expression in plasma cells. Then we have to bulk-lyse the sample upfront to concentrate the number of cells. So we need to add an extra step to our sample processing protocol. We need to make sure that our MRD panels are optimized for detecting the target population at very low levels, and that the panels are adapted to immunotherapies that can interfere with the expression of certain markers. And definitely, we need to make sure that our instrument has the capacity to collect a high number of events relatively fast. One of the biggest challenges in MRD assays, as we know, are the false MRD negatives, because we are handling a large number of events, and we want to identify a population that has a very low frequency. So high sensitivity is key to avoid reporting false MRD negatives here. Plus, we need to consider the patient treatment for the gating strategy. Some immunotherapies can mask the expression of certain markers. On top of that, in every pathology lab, we are getting more and more cases. We have a very tight turnaround time. Because of the COVID-19 pandemic, hospitals may have cut the budget for the lab. And at the same time, we need to meet all the regulatory requirements for diagnostic labs. So we can summarize all this in one sentence. In a clinical lab, we have to get results out quickly without compromising quality. Now, before introducing a new test in the lab, we need to carefully design and clinically validate the assay. And to design a new flow test, there are several aspects we need to keep in mind. First of all, the clinical question that we want to answer. And the clinical question is going to determine the panel design, the sample processing protocols, the instrument settings, the data acquisition parameters, and the data analysis strategy. Once we come up with the antibody panel, we need to test the entire assay. And this is an iterative process that can take many, many rounds of design, redesigned before we complete it. After we are done with this testing phase, we need to ensure that our assay complies with the current in vitro diagnostic regulation. And we need to put together a fit-for-purpose validation plan and assess several parameters like specificity, precision, sensitivity, which is essential for MRD assays, carryover, and even establishing reference ranges. Just to let you know that to date, there are no specific guidelines for validating flow cytometry tests in regulated environments, which makes the entire process more complicated. The question is there a way we can get consistent results over time across instruments and operators, save money and time while we comply with regulatory bodies? The answer to this question is yes, by standardizing all the steps in our clinical routine. And that's exactly what the EuroFlow method is about. The EuroFlow Group is an independent scientific foundation made up of over 20 companies and research groups, which was born in 2006 with the mission of developing, standardizing, and validating highly sensitive flow cytometry tests for diagnosis and follow-up. The EuroFlow method is a screening approach looking to use fewer antibodies at first to narrow down the malignancy, and those are the screening tubes, and then running classification panels for further differentiation. They started with leukemia lymphoma panels, followed by MRD panels to monitor patients. And once they got the system in place for oncomatology, they applied all their expertise and knowledge for the primary immunodeficiency and the immune monitoring fields. This EuroFlow method can be used in the oncomatology, clinical trials, and clinical immunology areas, but today we are just going to focus on the oncomatology. And this method includes the SOPs, the antibody panels, the analysis software tools, and a QA program. So now let's go over each of them. There is a protocol for bone marrow sample collection and transport, specifically for myeloma MRD testing. And also there are guidelines on sample collection for regular testing. What else? There is a protocol for sample processing and staining, including extracellular and cytoplasmic antibody staining guidelines. There is a specific bulk lysis protocol for MRD testing. For building leukemia, lymphoma, and myeloma panels, the EuroFlow tested a large number of fluorochromes and antibody clones, and they selected the fluorochromes based on the MFI, the stain index, the spillover, the stability, and the availability. Here we have an example illustrating the comparison between two fluorochromes, the APC-H7 and the APC-C750, conjugated with the same antibody clone, the CD10, and tested in the same sample under the same conditions. What we see here is the APC is that the APC C750 conjugate shows a brighter MFI, which provides a better resolution between the positive and the negative reference populations. Likewise, for selecting the markers, they looked at the antibody clones, the conjugates, the number of combined markers in a single tube, and the amount of antibody and volume of staining. They tested the performance of the marker combination together using software tools, and that's what we can see in these diagrams. So these are multidimensional plots showing cases of TALL in green, BCP-ALL in blue, and AML in red. And all these cases were stained with the EuroFlow acute leukemia screening panel. Now, in each plot, they left out one of the markers of the panel in order to study one by one its discrimination power of these three acute leukemia subtypes. And these results indicate that each individual marker included in this panel is essential because missing one of them affects the identification of the different acute leukemia entities. Now, there are protocols for instrument setup, calibration, and compensation available for different instruments, for FACSCAN2, Navios, and FACSLyric as well. And these protocols cover the instrument configuration, the setup of forward and side scatter, the setup of PMT voltages for target fluorescence channels using rainbow beads, the compensation settings, and the instrument performance monitoring. For the data analysis, we need flexible and advanced tools that make the multicolor analysis easier. For example, by turning off the background so we can just focus on the target populations, by looking at our data in many different ways, and by being able of studying the cell maturation path of a specific population. Also, we need tools to simplify the multi-parameter analysis. As flow cytometry technology evolves, there are more and more parameters to analyze. In this slide, we have all the possible combination of markers in an 8-color panel, and that's a total of 64 diagrams. And instead of looking at all these 2D plots, we can convert all of them into one multidimensional diagram that separates out populations using all the parameters in our panel. And this way, we can discriminate populations with a very similar phenotype that can go unnoticed if we just look at the 2D plots. We can check the complete immunophenotype of a target population versus a normal control. And we can follow up patients with confidence by loading the diagnostic population onto the MRD case as a reference. We can analyze patient cases and not single tubes, which is very important, because we can combine all tubes from the panel, analyze them together, and create one single report. Okay, still the problem in flow cytometry analysis is the lack of a good reference point or baseline. And as complexity of flow panels and data analysis increases, we really need big data analysis approaches. So we did automation for our clinical flow analysis. To put it simply, the automation consists of clustering all the events in the file, followed by an identification of all these clusters. But to be able to give the populations and clinical and biological meaning, we need to feed these algorithms with human knowledge and expertise, and that's the EuroFlow reference database. Okay, let's see an example of automation in acute leukemia screening. So we get all the cell subsets in our sample identified and characterized. And also we get a compass diagram pointing out the cell lineage, the T, B, or myeloid lineage, that the aberrant cells are more likely to belong to. This is basically done through a multiparametric comparison of your sample versus a reference database. And that reference, the reference database in that particular case, contains B leukemic cells in green, T leukemic cells in blue, and myeloid leukemic cells in red. Here we have the interpretation of the case, including not only the blast information, but also the detection of potential related disorders of the patient. And we get the complete immunophenotype of each of the aberrant populations in case that we have several lineage affected. And additional information on the present or missing populations of the sample. And what is more important, we also get information on which panel to run next for further classification of the disease. Okay, that's for the acute leukemia screening. Now let's see another example of automation in myeloma MRD. So here we can see the identification and characterization of both normal plasma cells and abnormal plasma cells after automation. And here we have the interpretation of the case, including a complete immune profiling of the patient versus a normal reference, information on the bone marrow regeneration, the MRD level and the immunophenotype of the aberrant population, the clinical threshold that determines the sensitivity of the test, and information on sample quality issues like hemodilution and sample viability. Okay, going back to the steps of a flow cytometry assays, EuroFlow runs 2 external quality assessment programs to certify the consistency and reliability of results generated in the lab. So we have the LST QA that monitors the overall flow testing performance, including the sample preparation, the instrument settings, and the data acquisition. And the MM MRD QA that specifically targets the data analysis of the myeloma MRD assay. The EuroFlow goal is to provide a standardized and validated, highly sensitive flow cytometry test for diagnosis and follow-up. And that requires a close collaboration between the industry and the academia. So thanks to this collaboration, Cytognos can bring the EuroFlow method as a ready-to-use flow solution for any lab in the world. And this is what we call next-generation flow solutions, as they include the laboratory SOPs, the dry-down antibody kits, the Omnicyt instrument, the Infinicyt software package, and ongoing training and support to help you along the way. To wrap up, these are the key takeaways from the webinar. Flow cytometry is a very powerful and versatile tool in clinical practice, and yet complex. Standardization is the way to obtain reliable and reproducible results. And the EuroFlow method is available through Cytognos as a ready-to-use solution for your lab. Just to let you know that if you have any question or you want more information about our next generation flow solutions, feel free to reach out to us anytime. And that's all. Thank you so much for watching.
Okay. So if you guys have any question about any of the previous sessions, we can answer them now. If not, we are going on a small break until the next session, which will be at 3 o'clock. Okay. And it's regarding, again, Omnicyt and also MMRD, since this is the main subject today, how to reach high sensitivity. And then at 3:30, we'll have a new session about T-cell immunotherapy research, which we are going to use a tube that was recently developed, which allows you to study the different T-cell subtypes. And then we'll have Ask Me Anything session, which will be live about the EuroFlow panels. Okay. Tomorrow we also have another session about COVID, multidimensional and spectral analysis using Infinicyt, another live session about Infinicyt, the Omnicyt online demonstration, and Ask Me Anything session. And finally, we'll finish with the presentation about the Omnicyt. Okay, so we'll have a short break now, and then we'll come back. And if you have any questions, just type them in the chat box. Thank you, Lorraine, for your comments. And we are here if you want to ask anything. Hello, so we are going to start our next session about the Omnicyt to achieve greater sensitivity with the MMRD studies. We hope you enjoy it.
Hello, and welcome to our webinar, Discover the Difference of the Omnicyt. My name is Dan Thacker, and I work as a flow technical and product specialist for Cytognos, covering the whole of UK and Ireland, and I'm going to be the presenter of this webinar today. So, let's start and move on to see our agenda for today. Firstly, I will start introducing Cytognos, who we are, where we come from, what we do, and then I'll talk about our unique NGF approach. From this, I will highlight some of our common laboratory issues faced by the best of us, and subsequently, how the Omnicyt aims to tackle and provide solutions to these factors. Finally, at the end of the webinar, if you have any questions, I will provide contact details so we can assist you with any of your queries or quotations that you may require. So, Cytognos is a biotech company from Spain that offers flow cytometry solutions for oncohematology and immunodeficiency fields. We are now also involved in large-scale national and international clinical trials. Our aim is to improve the efficiency and workflow in the laboratory, whilst equally enhancing patient care through our standardized, efficient, reproducible, and highly sensitive solutions. We are a clinical flow cytometry manufacturing company, and as you can see from our timeline, we were first created back in 1996 within the hematology department of the University of Salamanca in Spain. Thus, we have been around for nearly 25 years. In 2006, we started to collaborate with the EuroFlow group in bringing its validated flow cytometry methods to the market with exclusive antibody panels and the powerful analysis software. One major milestone we achieved is that we incorporated a CE-IVD certified instrument in our total flow solutions called the Omnicyt, while the most recent updates include the development of the PID tube for immunodeficiency detection, as well as, very recently, the inclusion of 21 CFR Part 11 compliant tools in our analysis software, which allows you to have increased data security and transparent audit. We have numerous offices ranging from in Boston, USA, to Rotterdam in the Netherlands, and also in the UK, while our main headquarters lie in Salamanca, Spain. We strongly believe in knowing each other leads to great mutually beneficial relationships, so here you have Chris and I, your friendly Cytognos staff, at your service. However, just to get an idea of our customer base, Cytognos is very much a global company, with products purchased all around the world through direct sales and distributors. Currently, we offer technical support to over 54 countries in the world. So, what is NGF, I hear you ask me? Well, in essence, it is a principle that covers a complete solution from start to finish. This covers the pre-analytical, the analytical, and post-analytical aspects of flow cytometry, which were developed and validated by the EuroFlow group. For those who are not as familiar with EuroFlow, they are a scientifically led European group of clinical flow cytometry experts, composed of multiple institutions that work together to provide best practice and a standardized approach to flow cytometry. They have been working for over 15 years now in the development and validation of a complete method to screen, classify, and then monitor diseases. All the EuroFlow publications are available online with open access, so you can check them out at the EuroFlow and Cytognos website for further information. Okay, so this is what the EuroFlow NGF method looks like for HemOnc, a method that has been proposed and evidenced through its publications. This approach consists of three major steps. So first, we have the screening step to determine any abnormality, what lineage might be affected, as well as the maturity level. From that, we have the second step, which is the more specific classification of disease. And finally, we have the third step of monitoring presence of any disease post-treatment. Here we have created highly sensitive panels to determine minimal or measurable residual disease, commonly known as MRD. So depending on the clinical question we want to address, we can choose different tubes and the consequent pathways. Therefore, Cytognos, in collaboration with EuroFlow, offers a complete and scientific-based solution to cover all the needs of a clinical laboratory that include the antibody reagents and dried panels, the EuroFlow standard operating protocols, the Omnicyt flow cytometer to acquire high-quality data, and of course the Infinicyt software to perform automated gating and analysis. All of this together is offered with an excellent technical support and training required to implement the NGF method. All of our NGF solutions are based on scientific and evidence-based EuroFlow research, so in that sense, what we do is bring the EuroFlow method to market. We make it available and ready to use for anyone to implement. So now, let's take a look at some of the common laboratory issues that I myself have been through, and no doubt some of these will be familiar to many of you. So firstly, no time. Nobody has any time anymore. Everybody is busy and multitasking is now essential, not just desirable. As you complete one task, no doubt another two keep popping up. Staffing problems. A lot of laboratories feed back that either they are understaffed and, if luckily, adequately staffed, but still face regular national changes in the de-skilling of the staffing pool. Staffing is one of the largest costs, hence management of change and other cost-cutting exercises do take place often. So automation is now really a necessity. It is one thing to have a body there, but if it takes more than resources to constantly monitor, often correcting repeat errors, then this is not really efficient. More and more, it takes additional time and real exposure to really gain experience to train, learn, and develop successfully. Finally, if you do eventually get to a stage where you do somehow have spare time, you have the staff available who do want to learn and also are capable, but only then to have an analyzer that keeps breaking down, having vacuum errors, unstable lasers, therefore constantly compensating, eventually leading to having to call engineers multiple times, who ironically are more present in the lab than some of the staff. Well, let me show you the solution and offer. So, I would like to present to you the Omnicyt flow cytometer, which can be the answer to all your problems. So let's look back at some of those common issues I mentioned earlier. With the Omnicyt, we can actually save time. The Omnicyt has a rapid startup facility, so there's no waiting around for lasers to warm. It has super fast flow rates that are adjustable for high throughput, and this is particularly great for MRD. You do not need to have lots of staff waiting around. The Omnicyt has multi-well sample processing facility with an auto sampler, and staff can use the Infinicyt analysis software from anywhere remotely, using automated gating, so subsequently freeing up skilled staff to be doing other essential tasks such as document control or audits, especially for UCAS visits. Skeleton staff can also be used, meaning enhanced service hours during the week, for example, or even on weekends for urgent service. Having equipment that is easy to learn and easy to follow, as it is all standardized, can make it easy to reach competence. The Omnicyt has a simplistic setup, and EuroFlow already provide all the standardized SOPs for the test to avoid day-to-day variance and minimize other complexities. We at Cytognos also offer training packages for staff, including visits to the head office and meeting with renowned experts, as well as the technical and support teams at your beck and call. Solid performance means no need to regularly compensate, no vacuum errors as it's volumetric, and the lasers also have an auto shutdown facility, and there is less need for engineers to visit. All of this indicates a more service resilient machine with greater sample processing time. It's worth knowing that we have great industrial links and business partnerships, such as with Thermo Fisher. This allows for fantastic developments to be made to utilize a CE-IVD certified analyzer, whilst also keeping on top of any customer demand due to its extensive manufacturing capacity. These relations mean being in top of new findings and the applications of latest technology incorporated into the equipment. Thermo Fisher will provide excellent and rapid engineering services with various packages available, whilst Cytognos can equally assist with the technical support. Cytognos has great experience in the hematology-oncology field and is supported by the EuroFlow research. Together, though, it is the end user that has all these benefits available to them. So, coming to some of the more specifics then, the Omnicyt is a 3-laser 13-parameter analyzer with the option of even further adding a 4th laser going up to 16 parameters. It has many unique and innovative features, making it ideal to work with the EuroFlow panels. Some of these features include having a flat top laser, for example, and utilizing acoustic focusing, which guarantees the most reproducible and accurate fluorescence measurements. At the same time, it reduces the frequency of system calibrations. There is no delay at startup waiting for lasers to warm, and equally with the simmer mode facility, the lasers switch off automatically when they're not acquiring or being used, hence it increases the laser life. It's a non-pressurized volumetric system that allows counting for cells in every population. The electronic system is capable of saving 20 million events, including all parameters, which is particularly important for MRD cases, and there is no abort rate. Therefore, all events are saved, including aggregates that could contain pathological cells. Again, really important for MRD. I have put this slide in to really highlight just how small and portable the analyzer truly is. As you can see, there are no chunky fluid carts, and as we know, a lot of laboratories just do not have the additional space for big, bulky, heavy equipment anymore. A key difference with the Omnicyt is the optics. The Omnicyt utilizes flat top lasers which are extremely stable. As there is no intensity curve with a maximum, even though a laser could get misaligned, there is no problems because the cells will continue to be excited with the same intensity. This is truly advantageous, as if the settings are so stable, you just do not need to keep redoing them so frequently. Hence, less engineer visits are needed. We also use the well-known rainbow beads to establish the targets, so once done, there is no need to mess around anymore. Settings, however, can be easily overwritten and are interchangeable between panels if you need to do so. Here then is a visual representation of quality control diagrams which quite clearly illustrate just how low the CVs are and how robust the analyzer is when compared to one of its nearest competitors. The superior quality just really speaks volumes for itself. As you can see here, these are the laser settings and the parameters in the Omnicyt, along with the dyes and colors that can be used. You additionally have the option of obtaining a 4th laser to be installed should you want supplementary colors, and hence can future-proof your technology for when more complex panels are released and implemented. The Omnicyt uses acoustic-assisted hydrodynamic focusing, thus avoids the compromise between data quality versus high sample flow rates. It achieves this by decoupling the cell alignment from the envelope liquid flow. This system uses an acoustic radiation pressure to transport the particles to the center of the sample flow, and this pre-focused stream is then injected into the flow of the flow chamber. This then produces a narrow flow of particles and uniform laser illumination. despite the sample entry rate. Therefore, the clear benefits are greater reproducibility and consistency of data while maintaining a reliable concentration results at all speeds, consequently obtaining a more homogeneous population. Thus, you could process either dilute or highly concentrated samples whilst still maintaining the low CVs. Talking of great reproducibility then, the analyzer even has a bubble detector facility that prevents the quality of the acquisition from deteriorating. There is also anti-clogging technology when working with more viscous samples. In addition, in the cases of MRD samples, the way the sample is actually dispensed in the flow chamber means that the events in the acquisition time do not drop as often happens in conventional cytometers. It acquires the sample and then drops it into the flow chamber. Hence, in essence, the first thing the system takes is the last thing it discharges into the flow chamber. Just like unique features on the fluidics and optics, the electronics too have special characteristics, such as the fact it can have more than 20 million events which can be recorded in a single file. With the 11 parameters and area, height and width, if some of these parameters were to be removed, it is possible we can reach even more. Interestingly, unlike its nearest competitor, there are no aborted events. All events are recorded, which is a key important feature for MRD assays. Finally, the resolution is 20 bits, unlike 18 on many of the other analyzers available, so a scale with more than 1 million channels to represent the data. Continuing with the electronics then, I've just put this slide here so you can really see what the startup screen looks like. It's very simplistic, it's not cluttered with fancy graphics or any impractical tools that you do not really need on a day-to-day basis. There is a rapid quick startup, so preheating of lasers is not required as they automatically heat up during the fluidic startup anyway, which lasts less than 5 minutes. The shutdown too can be adjusted to your needs, ranging from a 10-minute to a 60-minute clean depending on how deep a cleanse you require. You can even program the shutdown after your last sample, so staff do not need to be waiting around to close the machine. So, this is the main screen when you're processing a sample on the Omnicyt. Here you have the plots in the center with settings and compensation on the right, all with drag and drop and overwrite compensation matrix facility. Whilst the stop and start acquisition is found here on the left with event counts as well as how much sample and time is left. So, how is everything I have mentioned so far? Sounds really amazing, but how does it really translate into something more tangible that you can actually gain in the lab? Well, let me just take a second to talk about the sensitivity, for example. This is vital and important for true MRD detection, as the number of events that we collect has a direct impact on the sensitivity of our assay, and therefore on the quality of our clinical reporting. High event counts are needed in order so that we can talk confidently about MRD positivity or negativity when the frequency of abnormal cells is statistically relevant when considering both the LOD, which is the limit of detection, and the LOQ, which is the limit of quantification. As we can see in these diagrams, although this is the same sample, when we collect 100,000 cells right at the bottom, we cannot see any real cluster of cells. However, as we move along to 1 million events and then 5 million events, you can identify better the clonal cells and thus have confidence in your results, since we know they are above the LOD and LOQ. Therefore, as defined by the IMWG consensus criteria, The term MRD negative can only be used when following the EuroFlow NGF method. So this is the auto sampler I mentioned earlier that can be added to the flow cytometer to process high number of samples through multiple plates or tubes. As you can see, some of the features include having many different types of plates, to having auto cleaning facility, as well as variable mixing methods to minimize errors and maximize sample consistency. It's important to know as well that the Omnicyt is compatible with barcode reading and data transfer is possible via LIMS. Additionally, using Infinicyt analysis software can allow for data to be saved either as PDF or Excel file formats, for example, as well as CSV. So, in summary, this is an example workflow of a complete start-to-finish NGF solution proposed by Cytognos and validated by EuroFlow. It standardizes all the procedure, starting with the staining of the samples with the dried kits, together with the preset EuroFlow SOPs. These are then acquired with the EuroFlow compatible Omnicyt flow cytometer and then analyzed using the advanced Infinicyt software. This generates a clinical report for the use of medical consultants and laboratory staff. As you can see here, here is a quick summary of the unique key points of the Omnicyt that I have described previously. These include having special flat top lasers as well as using acoustic assisted hydrodynamic focusing to achieve high stability and sensitivity. Other key points include not having any abort rates and having an electronic system capable of more than 20 million events. Last but certainly not the least, we have an amazing support and technical team that offers assistance as and when you need, all the way from start to finish. It covers all the pre-analytical, analytical, and post-analytical aspects of flow cytometry. We aim to offer personalized support as suited to your own individual needs. So finally, I would like to thank you for your time, and if you have any queries, please do not hesitate to contact either me or us as the support team on the email addresses below. We really hope the Omnicyt can be the solution to your laboratory requirements. Have a great day. Goodbye.
Okay, thank you so much for attending this session. It's 3:23. We will now have another session starting in 7 minutes, more or less. Okay. Sorry, it's not working. Okay. Sorry, it's not it's not working now. I don't know why. But we will come back in 7 minutes, okay? No funciona. Okay, thank you for waiting. So I would like to go over the schedule for today. So we already saw the presentation regarding the Omnicyt flow cytometer. And now at 3:30, we will start our last recorded session, which is about T-cell immunotherapy research. And this is a presentation about a new panel that is presented to study all the different T-cell subtypes, maturation subtypes, which might guide you to study different immunotherapies. Okay. So the presentation will start in about 1 minute. I would like to present also the schedule for tomorrow. So tomorrow we'll have a short presentation about Cytognos so that you know us better and what we do. We then have a recorded presentation about the immune monitoring of COVID-19 patients using the PID Orientation Tube. Then we have a session about multidimensional spectral analysis with Infinicyt. And we have a live session regarding Infinicyt. So if you have any questions, you can come back then and you can ask them. If not, we will also have some content to present to you. And then we have our last Omnicyt online demonstration and a session where you can ask anything about the Omnicyt flow cytometer. And then we finish up with a recorded session about the Omnicyt and how it can help to have high sensitivity studies for MMRD. So we'll start the last recorded session now, and we hope you enjoy it.
Hello everyone,
Wait.
This is Dan Thacker.
No?
Hello everyone, this is Dan
Not
Thacker,
this one.
Flow Technical and Product Specialist for United Kingdom and Ireland.
Okay.
Hello everyone, this is Dan Thacker, Flow Technical and Product Specialist for United Kingdom.
I don't know what's happening.
Hello everyone, this is Dan Thacker.
Okay, just a second, please.
Flow Technical.
No sé qué está pasando. Just a second, please. Wait.
Hello everyone, this is rate between if T cells become. Hello everyone, this is Dan Thacker, Flow Technical and Product Specialist for United Kingdom and Ireland. Hello everyone, this is Dan Thacker, Flow Technical and Product Specialist for United Kingdom and Ireland. For this session, I will be presenting our new panel for the identification of T-cell subsets. In this presentation, we're going to focus on the composition, the function, and useful applications for our novel flow panel. So let's then start with a quick overview of the memory generation process in T cells. In homeostasis, naive T cells emerge from the thymus and recirculate between blood and secondary lymphoid organs, such as the spleen and lymph nodes. Upon recognition of their corresponding antigens, presented by antigen-presenting cells, commonly known as APCs, naive T cells become activated and differentiate into effector T cells, which are going to migrate to peripheral and inflamed tissues. They collaborate with other immune-associated cells until the antigen is cleared. After the removal of the antigen, most of the effector T cells undergo apoptosis, whereas a small pool of T cells then turns into long-lived memory T cells. So then, in the peripheral blood, there are two main types of circulating memory T cells which can be found. First one is the central memory T cells. These are that are characterized by high potential of self-renewal and the expression of molecules implicated in the homing to secondary lymphoid organs. However, these central memory T cells lack immediate effector functions. The second type is effector memory T cells. These, in contrast, effector memory T cells produce effector cytokines such as interferon gamma, interleukin-4 and IL-17, and express proteins involved in the migration to peripheral tissues. In this graph on the screen, you can see that one of the most accepted models of transition from naive to memory and terminally differentiated T cells. The model proposes that dormant, naive and memory T cells turn into activated effector cells and progress through the maturation pathway. This maturation pathway is not always linear and cells can jump stages. During this transition process, cells lose some proliferative potential and lymphoid homing capacity, and they progressively become more cytotoxic and produce higher amounts of effector cytokines. At the same time, they gain tropism to inflamed tissues and senescence markers. So, after this brief scientific background, we can move directly into looking at the kit. It is an 8-color panel for the identification of different subpopulations of naive and memory T cells. CD45, CD3, CD4 and CD8 are used to identify helper and cytotoxic T cell populations, and equally we have included additional markers to classify CD4 positive and CD8 positive T cells into different subpopulations of naive and memory T cells. For example, CD27 is utilized in this panel, which is a co-stimulatory molecule whose expression is lost in effector cells. We also have CD45 Rho, which is expressed in memory T cells. And finally, we have CD62L, also known as L-selectin, which is expressed mainly in naive and central memory T cells. Last but not least, you may have noticed that we have left the FIT-C channel intentionally free for additional drop-ins. Customers may add complementary antibodies for the identification of more specific T cell subpopulations, or include FIT-C conjugated reagents for the detection of CAR T or antigen-specific T cells. Let's jump now into the main T cell subpopulations that can be identified with this kit. We will evaluate CD4 positive T cells just as an example. So here you can see naive T cells are characterized by high expression of CD27 and CD62L, however with dim or negative expression of CD45RO. Next then, we have the central memory T cells. As you can see, these have high or positive expression of CD27, CD62L and CD45Rho. Here we can see transitional memory T cells. These express slightly lower levels of CD27 than central memory T cells, and transitional memory T cells still show high expression of CD45Rho, but lose expression of CD62L. Here then you can see the effector memory T cells on the plots, and you can see that these are CD27 negative, CD45Rho positive, and they present variable expression of CD62L. Finally then, we have the terminally differentiated T cells, which are CD27 negative, CD45Rho dim or negative, and they show variable expression of CD62L. And this is then a summary of how CD4 positive T cell subpopulations look like altogether in two different Infinicyt dot plots. In the case of CD8 positive T cells, similar subpopulations can be identified in terms of the full immunophenotype, with the loss of expression of CD27 and the expression of CD45Rho in the memory compartments. However, an additional subpopulation can be found among the CD8 positive T cells that is not present in the CD4 positive T cells. And these are the early effector CD8 positive T cells. They are characterized by dim positive or positive expression of CD27 and negative expression of CD62L and CD45RO. Now let me just quickly show you some unique tools on our Infinicyt analysis software which are particularly useful when analyzing samples stained with the T-cell subsets kit. Firstly, a maturation database can be established with normal samples, thus enabling a quick overview of any abnormality in the sample with respect to the maturation diagram. In the same context then, a reference image can also be created with normal samples, thus allowing a fast comparison with an abnormal sample. So what is the aim of this kit and what can be the potential uses for this panel? Well, firstly, monitoring oncohematology patients, taking into account that memory T cells populations can and do change during a response to a tumour. Another useful aspect of this kit is the measuring of the response to vaccines or infections. In this context, the free FIT-C channel can be utilized for the identification of the virus-specific T cell subpopulation. Also, in a similar manner, the free FIT-C channel could also be used for the separation of CAR T cells from the endogenous T cell population. This is one of the most important intended uses for this kit. Now we will take a look at some examples of memory T cell studies found in the literature available online. In this first example, this publication evidenced and analyzed the distribution and function of the different memory CD4 positive and CD8 positive T cell subsets in patients with non-small cell lung carcinoma, often called NSCLC, compared with healthy donors. They used peripheral blood and lymph node samples. In this case, they utilized CCR7, which in terms of immunophenotyping is equivalent to CD27. They used CD45RA as a marker of non-memory cells instead of CD45Rho that is expressed in memory T cells. This publication and authors discovered that patients had an increased percentage of circulating CD8 positive T cells. In lymph nodes, NSCLC patients showed similar frequencies of CD4 positive and CD8 positive T cells, whereas in lymph nodes from patients with other types of cancer, CD4 T cells were more abundant than CD8 positive cells. The paper states that patients with non-small cell lung cancer present a different frequency of naive and memory T cell populations compared with age-matched healthy donors. Essentially, that they had more effector and reduced central memory, naive and effector memory T cells. They described that NSCLC patients had a reduced percentage of circulating interferon gamma positive, TNF alpha positive and IL-17 positive T cells. In this point, it is relevant to say that although the analysis of T cell memory immunophenotype is not part of the diagnostic or prognostic processes in oncology, it can be implemented easily to clinical routine settings as a simple and fast method to monitor the immune response to tumor and/or possible treatment and therapy. Here is another publication where the paper compares the frequency of CD4 positive and CD8 positive T cells in healthy donors and patients infected with the parasitic worm S. stercoralis. They also quantified the levels of certain cytokines in the plasma. The publication established that they did not find significant differences in the percentage of CD8 positive memory T cell subsets and found increased levels of IL-4 and IL-9 in plasma. Regarding the CD4 positive T cells, people infected with the worm Strongyloides, they present increased naive T cells and reduced central memory T cells. Interestingly, after anti-parasitic worm treatment, the frequency of those populations are reverted. In this publication, the response to COVID-19 is analyzed. The authors study the response of immunodormant and overall SARS-CoV-2 specific memory T cells in patients who have recovered from COVID-19. Peripheral blood mononuclear cells, or PBMCs, obtained from 42 patients and 16 unexposed donors were tested. The paper describes that severe COVID-19 induced a higher scale and magnitude of T cell responses. Virus-specific CD8 positive T cells are identified thanks to the labeled HLA pentamers. With this strategy, the paper compared the memory phenotype of COVID-specific T cells with unrelated circulating T cells. They found reduced percentage of naive and increased effector memory in COVID-19 specific CD8 positive T cells when compared with the unrelated T cells. Regarding this issue, it's important to comment that T cell immunity is becoming more and more relevant in monitoring COVID-19, general immune status, and also in analyzing the response to any vaccination programs. As highlighted previously, this kit is particularly useful for the monitoring of CAR T cells. CAR T cells are genetically modified T cells engineered to recognize a particular tumoral epitope. Receptor genes are transferred to T cells purified from peripheral blood and expanded ex vivo. Then, this product is infused again into the patient, leading to T cell mediated destruction of the tumor. CAR receptors are constituted by different domains. So you have the antibody-derived antigen binding domain. You also have one or more co-stimulatory domains, and you also have CD3 zeta activating domain. The field of CAR T-cell therapies has experienced a great shift in recent years. In 2017, clinical trials with CARs targeting at least 11 different molecules had been completed, including CD19, CD20 and CD22. From this day onwards, several additional trials have been carried out with the CARs directed against new targets, as emerging therapies for the treatment of both hematological disorders and solid tumours. The free FITC channel in our kit allows the addition of reagents for the identification of CAR T cells. There are multiple strategies to achieve this. So firstly, include an antibody that recognizes the structural domain of the CAR, or another option could be the use of the target protein of the CAR labeled with FITC. The advantage of this method is that the labeled protein can bind to all CARs directed to it, regardless of the structure. In the last few years, it has been proved that CAR T-cell products that are rich in central memory T cells are more long-lasting and more effective in tumor reduction and removal. This fact highlights the importance of our kit in characterization of T-cell products when generating CAR T-cells. As you can see, in the lower part of this slide, there is an example of monitoring of infused CAR T-cells over a period of time. So, After defining the multiple uses for the panel, it's time to briefly describe the kit format and its characteristics. The kit consists of 4 vials with lyophilized or dried reagents containing 5 tests each, for a total of 20 tests per kit. It also includes compensation tubes for specific reagents, which are the tandem dyes and, of course, the Cytognos exclusive fluorochromes. To summarize then, Cytognos offers a single tube 8-color panel which guarantees a simple application with increased reproducible results for T-cell subsets. The panel is easily adaptable to any specific study, as the free FITC channel allows the addition of FITC labeled antibodies or proteins. Furthermore, the staining and acquisition of samples is rapid, which takes less than an hour. Finally, our carefully selected antibodies and fluorochromes via evidence publications facilitates an accurate immunophenotyping of naive memory T cells, allowing stringent follow-up of all T cell populations. These features make our kit a valid and solid solution for the monitoring of immune responses to tumor and therapies, monitoring of CAR T cells, and evaluating responses to any vaccines. So finally, I would like to briefly thank you for your time and attending this presentation. If you get a chance, don't forget to check out our newly designed website, where you can find lots of new information regarding video tutorials and an academic section where you can have further education on diseases, as well as novel COVID-19 flow solutions. You can also get regular information through our Cytognos newsletter to keep up to date with next generation flow solutions, as well as the latest webinars and events. Thank you. We will now go to any questions.
Okay, thank you so much for attending this last recorded session. We will start a live session about the EuroFlow panels very briefly in about 9 minutes. So if you have any questions, please write on the chat and we will answer during this live session, okay? So the session will start exactly at 4 p.m.
Hello, hi everyone. We're back now. I hope you are fine and you are enjoying the ESCCA 2021 Congress. We are here again to enjoy the last session planned for today. Let me share my screen, which is the Ask Me Anything regarding EuroFlow panels. Well, of course, if you are there, you have any question regarding any other topic, please don't hesitate to let us know, and we will be more than glad to help you with any doubt or comment that you may have. As I said, you can write down in the chat if you want, or you can write to us through our private chat. And okay, so now I will do remember a reminder also that this is all the sessions are recorded, so you can come back to this later if you want, and also you can watch the sessions from yesterday or the sessions that will take place tomorrow. Tomorrow we'll repeat the immune monitoring session using the primary and deficiency orientation tube. We'll watch again the multidimensional analysis with Infinicyt. We'll have a live demo analyzing with Infinicyt, and then we'll have the Omnicyt online demonstration. As always, it will be at 12:30, and then we'll have the final Omnicyt multiple myeloma session. Will be available anyway in the chat as always. So yeah, don't hesitate to let us know. Let me visit here the calendar. On our website, you can go to events, upcoming congresses and meetings, and you can access here the information about the ESCCA. When the ESCCA is over, you will be able to access that here through show past events. So when you access here to show past events, you will be able to access here. Okay, and in this website you have information about the talk that we had this midday regarding multiple myeloma reporting or minimal disease assessment. Then you also can download one of our 6 brochures available. You can download them direct, like by filling in this form, or you can access those directly from our virtual stand in the virtual sponsor section in the ESCCA website. And you also have the schedule about from our sponsor room, so that you can watch this later. And you can request your private online demonstration in case you are interested in that. So if you want to have a private session with us where you can ask more like private information, you want to know the details, or maybe you want that Arantxa or Maria show you other parts of the machine, or you want to see some specific feature of the software, just request that on the site demo, and we will be glad to schedule that with you, and we'll have that session. And finally, remember that there is an ESCCA raffle where you can participate by filling in a few questions. We include a few questions that were quite easy in our opinion, of course. But if you have any question or doubt about those questions, just let us know and we'll be glad to help you. And you can win a full year Infinicyt license. So this will be very useful for those of you who know Infinicyt and who are using Infinicyt already. You will have an additional license, advanced license, it's important, including also 200 EuroFlow database access, so that you can try the database, you can run your own samples, and well, that's quite a lot of samples to run. Yeah. So now let me change to another aspect. In the same manner that I did yesterday, I would like to show you more about some of the EuroFlow papers, so that you can actually come back to them later. You can check them out on your own, and they will serve you as a training and an initial way, let's say, initial point to start learning about EuroFlow, to start learning about the research that they have been doing in the last few years, and how you can implement this methodology at your lab. Again, if you need more information or more details about this, you can write to us, you can contact through our website, and we'll be glad to help. So this is the initial publication of EuroFlow in 2012. And this was, all these publications in 2012 were included in a special editorial, or in a special chapter in the leukemia magazine. And in this first piece, they actually describe the EuroFlow consortium, which basically was a scientific group composed of laboratories, of research laboratories in different countries in Europe, also in America, because there was a lab in Brazil, and also two small medium enterprises. One of these enterprises was Cytognos, and we are proud to be one of the founders of the EuroFlow consortium. And the general aim, or the initial aim, of the EuroFlow project was to develop and standardize fast, fast, accurate, and highly sensitive flow cytometry for the diagnosis and classification of hematological malignancies, and of course also for the treatment and follow-up. During this first few years, so from 2006 to 2010, they performed this research by testing different antibodies by the different EuroFlow members. So they were evaluating different antibodies and different conjugates, fluorochromes, in the different centers. Both the research laboratories and us as company were testing different products in order to build the best panels. Then they also did other experiments, not so related to the flow cytometry, the EuroFlow panels as they are known. There were some additional studies or essays developed related to beads, immunobead, for detection of these fusion proteins. And this is part of the research that you can see there too, although we won't be covering that today. And then they developed these multicolor flow cytometry protocols and panels. That's what we are gonna talk about more. And they also focused on the creation of innovative or novel software tools for multidimensional analysis. So this is what Infinicyt consists. So this is they're talking here about the development of Infinicyt as a software, and how this new software, which we started to develop in 2006, actually became a reality. These are some of the labs that were part of the initial EuroFlow members in 2006, and then additional members later. As you can see, we have the lab of Alberto Orfao in Salamanca, Van Dongen in Rotterdam. We have Cytognos here, Lisbon. We have Kiel, Paris, Leeds, Prague, also Poland. Okay, so there were different members, of course, and they has been evolving throughout the years, and more members have joined EuroFlow in the last few years. After after these 5 years, they what the EuroFlow did was actually to publish all this information. So they started in 2006, 2012 is when they published this chapter in leukemia, and they actually not that one. And they actually introduced this paper, which is the article that includes the original EuroFlow panels for hematological malignancies. This is quite a long paper that covers the design, the creation, the design, the creation, and also some of the validation related to each of the panels developed by EuroFlow in this first phase of the project. Okay, so you can see here the screening tubes of the screening panels of EuroFlow, and then we have the characterization panels, and well, of course, the point is that the EuroFlow methodology consists on the fact that you are trying to standardize the whole approach to hematological malignancies diagnosis and monitoring. So based on some predefined suspicion, you will start by doing, usually in most cases, a screening tube, acute leukemia notation tube, lymphoid screening tube, plasma cell screening tube, and small sample tube that are whose objective is different kind of diseases. So if you have a high suspicion of acute leukemia, you will run the acute leukemia notation tube in order to find out what's the lineage of that acute leukemia, whether this is a B-cell, T-cell, or myeloid acute leukemia. And then, based on the result of this screening tube, you will run the characterization panel, any of those, that will be used in order to obtain the final diagnosis. You will be either based on the marker express, you will obtain a conclusion. So the disease is this one, a CLL, follicular lymphoma, whatever, or you will characterize the population. So this more in the case of acute leukemia or AML, where you will need to find out whether the population is positive for one marker and negative for other, for treatment purposes or for other clinical aspects. Okay, next. Basically, what they did in order to create all of these panels, as I said, and they mentioned in the first objective, was actually to find out the best markers and the best fluorochromes that were useful for this process. So actually what they did is that they had different markers and they started testing some of the markers. For example, in the case of the acute leukemia notation tube, they started testing some markers in an 8-color combination. All these EuroFlow panels in the initial phase, in the first phase, were designed for 8-color flow cytometry. And basically what they did was, okay, we will have this panel as a starting point. We ran, in this case, they ran 35 samples within different centers and different laboratories, so that they can actually test what's the performance of this panel and what's the validity of it. And then they will change the markers, or they will change the fluorochromes depending on the outcome of the result of the analysis. Sorry. So yeah, the point is that they didn't come up with a panel just because of their personal experience or because of their personal decision. They actually came together as a group of different laboratories. They proposed different markers, and they actually validated that those markers were the ones that were most useful for the diagnosis or for the discrimination between abnormal and normal populations. That's kind of what it's on here. So the point is that using Infinicyt, they actually compared by using the APS, the automatic population separator, they actually compared what was the impact of having one fluorochrome or not having it, sorry, not one fluorochrome, one marker or not having it in the panel. So in Infinicyt, you can actually say to the APS, please include CD3 in the comparison, or do not include CD3 in the comparison. So actually, by including it or not, you will be able to see whether the separation between the abnormal and the normal population increases or decreases. So basically with that, you can say, oh, actually CD3 is not helping us to differentiate abnormal from normal. So maybe it's not that useful compared to other markers. Anyway, remember that also there are like 2 types of markers that are included within the EuroFlow panels. And that's mentioned, I think it was mentioned in the other. Yeah, here. So there are 2 groups of markers that are included in the multicolor staining. There are some markers that are used for the identification of cell populations in the sample. So these markers allow to actually identify a population. So for example, if you are analyzing lymphocytes or you're analyzing B cells, you want to have their CD45, you want to have 19, and you want to have 20 there. If you're analyzing T cells, you probably want CD3 and you want CD7. So you use these markers in order to identify these cells, and these are the backbone markers. Okay, these backbone markers are usually, well, the backbone markers themselves are always present in the panel in the same position under the same, so they are present in all the tubes of the panel in the same position. So they are conjugated with the same fluorochrome. What's the point of this? That you can actually use the same identification strategy in order to identify your cells. So no matter whether you are using the first tube or the last tube of the BCP LD panel, the one for lymphoproliferative diseases, you will always identify B cells using the same approach. Then there are other markers that are the characterization markers, and these are used for the characterization of each population. So yeah, you want to know the phenotype of some populations, either to classify the cells in different subgroups, or in order to do the diagnosis, or to characterize some abnormal phenotype. So these markers are included in every single, in each tube, and they might be included in different positions. Of course, they are not repeated in the same panel because it doesn't make sense to repeat the marker in most cases. And yeah, and so that's the point. So basically, for the ALOT tube, for example, they came to conclude that they would design, that they would include all these markers, okay? So they have included cytoplasmic CD3, CD45, cytoplasmic LPO, cytoplasmic 79A, CD34, CD19, CD7, and surface CD3. If you read the paper in detail, you will see that they actually cover also the rationale between like in order to select some of these markers. For example, the fact that you want cytoplasmic and surface CD3 here in order to differentiate between T cell precursors and mature T cells. But well, you can have much more information there because of course there are always some markers that are not so common in other panels. So some people prefer one marker, some people prefer other. So usually in this paper, EuroFlow explains quite in detail the reasons why they selected one marker over another. So yeah, here you have the design of all these tubes that are included here, all of them, okay? And we could say that this is actually the first phase of the EuroFlow project and the EuroFlow panels. The other interesting paper related to this first phase, or one of the others, I mean, there are several papers included apart from these that I'm showing here, okay? But the other element that is very important in this first phase of the project is actually the EuroFlow standardization of the flow cytometer instrument settings. So, and also the immunophenotyping protocol, so the sample processing. So basically, the point is that when EuroFlow started to work here, they didn't want only that the labs were using the same panel. So that's already quite an improvement, okay? So if you are working with several labs, let's say around Europe, let's say it might be interesting that all the labs are using the same panel so that you actually analyze the sample in the same manner. You have the same kind of information, and you can actually combine all that data together in order to obtain more insightful data. Apart from that, the point is that actually, as you know, flow cytometry is quite a not so standardized method. It's quite open. You can modify the sample processing. You can modify the instrument settings. You can have different voltages in your instrument, and you can have different compensation. And at the end, all of that will have a huge impact on the outcome of your analysis. So you, maybe some people is used to having lymphocytes in very high site scatter and focal scatter, so they just see lymphocytes, and other people might want to see all lymphocytes, monocytes, and neutrophils. So all of that has a huge impact, not only on the analysis and how the lab works, but also on the final outcome of the research and potentially also diagnosis. So they propose here a very significant project related to the standardization of both the instrument settings and their sample processing. So basically, for that, they use initially the let's say the standard instrument that was used for this project was a fax Canto 2. Also, the Beckman-Coulter Navius was also included here. At the same time, they were doing both, or kind of at the same time. And what they did was that they selected what were the best fluorochromes that could be used for the 8-color panels. Again, this in the first phase, they were only 8-color panels. In the later years, just a few years ago, they started publishing also panels with more colors, up to 12. And well, one of the main aspects here is that in order to, apart from selecting the best fluorochromes, you have to make sure that in different instruments, let's say if you are using the fax Canto 2, if you are using one fax Canto 2 in one lab, another instrument in another one, you would like that the result it's quite reproducible, and you want that the cells actually fall in the same place. In order to do that, you cannot really use the same voltages, because the two instruments they are not exactly the same technically. The lasers might have a little bit of different power, so you cannot use exactly the same voltages. What you do is the other way around. Actually, you use a specific type of beads, and that's what EuroFlow identified as the rainbow beads, the EuroFlow rainbow beads that you run through the instrument. With the EuroFlow rainbow beads, EuroFlow proposed basically some target MFI values. So these are the MFI values that were used with one of the lots of the rainbow beads at that point in 2012. So basically, in your instrument, you run the rainbow beads, you acquire them, and you make sure that the result that you obtain for these rainbow beads is around this number. So you modify the voltage of your of each of the fluorochromes in order to obtain this MFI. By doing that, what you do is that later when you acquire the sample, if you acquire the sample in two separate instruments, the result, the CD45 expression, the CD4 expression, the CD10 expression, CD20 expression, they will be very similar. And this is a way how they do it, okay? They, of course, here they also explain the rationale behind why they chose these rainbow beads, how they actually adjust it for the best position, and basically depending on the PMT voltage that you have to use, and the standard deviation and the coefficient of variation of both of the instruments in general. So yeah, this was quite a deep process. And then also, so that's we already set up the fluorochromes. We set up the instrument configuration, meaning voltages, and then we also had to standardize the compensation, of course. And for that, there is also additional information from that side. For example, in our kits, in the Cytognos lyophilized kits, we include the specific antibodies that you need in order to perform the compensation. So we are providing those to you. So actually, when you run our tubes, you only need to acquire our antibodies, the one that we provide in the kit for you, so that you have exactly the same antibody that is included in the kit. In that manner, the compensation should fit exactly the sample that you run later, and there shouldn't be any problem there. And then the last step that I wanted to mention too, sorry, there's a lot of light here. The last step of this process that I wanted to mention is actually that they also focus a lot in the sample preparation. Of course, different labs were using different processing methodologies: stain, lyse, no wash, stain, lyse, wash, stain, lyse, wash, fix. And this has an impact on the cell, on how you see the cells, especially regarding scatters, the 4-1-side scatter, but also on the expression of the markers, okay? And then there were other, the result also depended on the lysis solution used. So that could be in-house lysis solution, or it could be commercial lysis solution. And some of them, that the ones that they tested were ammonium chloride, FACS lysis, quick lysis, and VersaLyse. FACS lysis BD, quick lysis, Cytognos, VersaLyse, Beckman Coulter. And well, basically they run different tests, and what you can clearly see here is that they are different. There are different results here, and indeed the FACS lyse with the stain lyse wash seems to provide a very good result in terms of separation of the populations, lymphocytes, monocytes, neutrophils, and eosinophils. They are quite together, the cells here. You can differentiate between them, and this is robust across different samples. And there is no need to fix the sample afterwards, which has an impact on the expression of some markers. So yeah, this seemed to be the most appropriate sample processing. Of course, for example, later when they went deep with the multiple myeloma, MRD, the minimal residual disease, in minimal residual disease, you want to have a high sample concentration. So at that moment, what they actually proposed is that you process a sample first with ammonium chloride, what we call the bulk lysis. And with the bulk lysis, you can process up to 2 ml of peripheral blood or bone marrow, and then you can concentrate the sample in a few microliters, and then you can acquire up to 10 million cells. So here with FACS lysis, we are talking about a few 100 to 1,000 cells, usually processed, and then in the minimal residual disease, in the high sensitivity studies, you want to process much more. So yeah, here you will see quite a lot of information, and just another element that I would like to show you. So all of these papers are or can be accessed from our website. Just, I mean, if you cannot find them in our website, you can go to Academy, Cytognos Technology, EuroFlow, and there in the resources at the bottom of the page, you have access to these publications, which are open access again. So you have the initial publication, the one with the panels, and the one with the technical instrumentation and the technical details, let's say. If you go also to the EuroFlow website, and here I will check the EuroFlow SOPs. The EuroFlow SOPs are published by the EuroFlow Consortium on its website. And actually, if you click there, well, you can find the new EuroFlow website, which was updated a few months, I think, ago. And actually, when you join the EuroFlow website, you can create an account for free. Okay, so if you don't have an account, you can create that for free, and there you can access all the EuroFlow protocols. So you have access to the rainbow bead target, the MFI that you need to achieve with your instrument, and you have different SOPs for sample preparation, instrument setup, and compensation for the different instruments you see. For this is for Canto, Lyric, Fortesa, Navios, and how to perform the backlysis, guidelines for bone marrow sample collection, and also you have more information about the panels. So the panels, yeah, here, composition information of the EuroFlow panels for hematological malignancies. If I open that, just for you to see that. You see we have again that beautiful poster, and there you have the different panels. So the next, that's kind of the first phase of the EuroFlow project. And then what EuroFlow did was to develop the, let's say, monitoring panels. The first of these publications related to monitoring was this one. This is a publication of the multiple myeloma MRD panel, next generation flow for highly sensitive and standardized detection of minimal residual disease in multiple myeloma. So this was published 5 years later after they performed this research, of course. And well, in here, they actually published for the first time the multiple myeloma MRD panel that consists on 2 tubes. Here you have the final version, so the ones that I selected here. This is the final version of the multiple myeloma MRD panel. And well, you see, well, there's something that I forgot to explain. Sorry, I forgot to explain before related to the backbone and the characterization markers, because I wanted to show you. Let me find the one of the big panels, for example, the TLL panel here. Okay, so I just wanted to say that in the panels by EuroFlow, I already said that the backbones are present in all the tubes, but the characterization markers are only present in one of the tubes. So that's what you can see here. This is a TLL panel, so the panel for analysis of T-cell acute leukemia, and you can see CD3 cytoplasmic is present in all the tubes, CD45 present in all the tubes. Then we have a series of markers in different fluorochromes, and then we have surface CD3. So basically, that allows you that you can identify T cells in all the tubes using the same strategy, so 45, CD3, and CD3. But you can use each of the tubes for the analysis of different well, markers. So basically, depending on the marker that is positive and the ones that are negative, you will come to a final diagnosis. I wanted to explain that because a similar thing happens here in the multiple myeloma. So we have several backbone markers that are used in this case for the identification of plasma cells. So 138, 27, 38, 56, 45, and 19. All these markers are used for the identification of plasma cells. But then we have two characterization markers, 117 and 81, that are used to characterize the abnormal plasma cells. Plasma cells are usually normal plasma cells. I mean, they are CD117 negative and CD81 positive. So with that, you can characterize them. And then you have kappa lambda in order to assess the clonality. You can see here that they point here the ME. This is actually the multi-epitope antibody. So this CD38 multi-epitope, which is one of the antibodies that we have available in our catalog, and the important feature about the CD38 multi-epitope is that it provides well, it can target different parts of the CD38 molecule, so that actually even under the presence of treatment or after treatment of the patient, you may obtain a higher signal for CD38 compared to other monoclonal antibodies, where after treatment with anti-CD38 treatment, such as daratumumab, you will lose that expression. So basically, it's quite difficult to identify the plasma cells in these treated patients. Here we had an example that I can show you about the how the panel is used. So you see here where this Infinicyt analysis with Infinicyt, and you can see how they use Infinicyt to identify plasma cells, normal plasma cells in blue, abnormal plasma cells in red, and well, they are in the normal area for plasma cells, 38, 138 positive, so plasma cells. Normal plasma cells are 45 positive, 56 negative. I would like to tell you that still, when EuroFlow started acquiring so many parameters, so many events, up to 10 million, up to 20 million, even more than that, what you see, I mean, you are increasing the sensitivity quite a lot there. So you are acquiring a lot of events that sometimes or usually you wouldn't see in your sample. You wouldn't see them not because they are not there. These cells are always there in your sample, but you cannot see them because there are not enough cells so that you can see them. So something that I want to tell you is that actually we realized, and EuroFlow realized before, that there are some normal plasma cells also that can be CD45 and negative. Okay, so that's something that happens, and it's not really, it's not necessarily a marker of abnormality. In this case, of course, this plasma cell is not only 45 negative, but also 56 positive. Then going to the next diagram, they are also 19 negative, 81 negative. They are 170 negative, so they are kind of normal in this area, a little bit less, 27. So that might be abnormality, but this is quite normal. And then they are lambda positive. So you see this why there is a clonal population. In Infinicyt, you remember that these are two tubes, right? So tube 1 and tube 2. In tube 1, we only have 117 and 81. In tube 2, we have kappa lambda. In Infinicyt, that analysis, you do that together. So here we have the two tubes together, two tubes, and then only here we only have one tube and one tube. So this tube will allow us to see the abnormality here, and this tube will allow us to see the clonality. But in the other tubes, we are combining both. Well, in the paper, in this research, they always compared the next generation flow approach to the conventional flow, and they actually found that the next generation flow was more sensitive, and it was able to provide a better diagnosis of, well, not diagnosis, but monitoring of patients compared to conventional flow. They also compare, you see here the sensitivity, and you see how in the next generation flow you can achieve up to 10 to the -6, depending on the amount of cells that you are acquiring, whereas in the conventional flow, this could be around 10 to the -4. So, well, all of these samples, basically you are losing them there. You cannot find them. So you are saying that the patient is negative when he is not, he or she is not. And well, they also compare that with next generation sequencing, and there was also a significant correlation between both, and that's described here in the paper much more in detail. Okay. Related to this, a few years later, they actually, well, the EuroFlow consortium, and I'm talking about different people here. Okay, so this is there are several groups involved in all this research in the same manner that in all of these papers. And the thing is that they actually published 1 year later. So this was 2017, 2018. They published their approach to use next generation flow for multiple myeloma for the minimally invasive characterization of NGAS and multiple myeloma, basically by analyzing circulating tumor plasma cells, what we call CTPC, in peripheral blood. For that, they are using the same panel. So they're using the panel that I described before with 2 tubes, Kappa Lambda 117 and 81, and they analyzed well, more than 500 samples, peripheral blood, and their pairs in bone marrow. And basically, they did a comparison between the outcome of the bone marrow result with the outcome of the peripheral blood result. What they found is that actually the measurement of CTPC here, it could be used as I said, as a non-invasive tool to assess the presence of plasma cell, abnormal plasma cells potentially in the bone marrow, because actually the presence of peripheral blood circulating tumor plasma cells was clearly associated with malignant disease in the bone marrow. Much more details about that in the publication, of course, if you want to take a look at that in detail. But yeah, these are very interesting paper and very recent research. So if you want to know even more about that, I'm also sharing with you here the fact that we have a webinar available related to that in our website. So if you go to events, recorded webinars, here you can access all of our previous webinars. And we have one given by Dr. Paula Tavares from Würzburg in Germany, that she's already implementing this kind of analysis in clinical routine and, well, this clinical research diagnostics in multiple myeloma. So you can watch the webinar and download more information about the presentation there. Okay. And then, yeah, during the day we have been speaking about the fact that in our automatic gate identification tool we include a consensus report and a standardized report that is based on the scientific consensus. So basically, some of the information that we include in the report is coming from several scientific publications, such as this one, for example. This is a publication by the International Myeloma Working Group, and this is already 2016, so before the multiple myeloma panel by EuroFlow was published, and they actually cover what was the state of the art for multiple myeloma diagnosis a little bit, I think, but also especially the assessment, the minimal residual disease study by different methods. So they are talking about the conventional flow, they are talking about next generation flow and other methodologies that are useful. So these are very insightful paper also if you want to know more about the history behind all of these papers and publications. Now I would like to move on to the other side of the EuroFlow research, and this is basically the immunology research that they started, or they kind of published a few years later. Okay. So basically, this is it. This is the first publication that covers kind of the This is a publication that includes like the whole, or describes the whole EuroFlow methodology for the study of primary immunodeficiencies and immunological studies by EuroFlow. So basically, the aim of EuroFlow here was kind of the same that they had been doing before for hematology. So they wanted to provide a solution to a standardized clinical analysis by flow cytometry of primary immunodeficiencies. And again, the approach was similar to the one used in hematology. So they use one orientation tube, which is the PID, the primary immunodeficiency orientation tube, for the screening of the samples, so that you can actually assess whether some populations, some big populations are missing, whether there is something strange, and it can serve as an orientation, as the name says. And then you can use other panels that go after that one in order to characterize the population, or characterize the disease, or do the final diagnosis. So based on your suspicion, you will run some screening tube or some panel, and finally you will come to a diagnosis. As you saw, this was published just in 2019. Okay, and here in this publication, they include all of these panels, as I said, the PID, the one for T cells, the one for B cells, the different type of tubes, pre-germinal center, post-germinal center, everything. And an interesting thing that they did here, of course, this is a piece of research that took a significant time for them to do. But a very interesting thing that they did here was actually that they had hundreds of samples of normal healthy donors that they actually analyzed with some of these panels. So for example, with the PID tube, they took all of these samples from healthy donors from different ages. So you see here cord blood, newborns, a few months, up to more than 60 years. They took all these samples and they measured them, and they analyzed the populations and the frequencies of each population present in these samples. With that, basically they built like this reference and this reference database where you can actually see what's the expected expression of the expected frequency of each population depending on the age of the patient. Right? So this, of course, the immune status of the patient or the people in general is not the same through all ages. It changes when we grow older, and yeah, basically that has an impact on the diagnosis of the disease. It's not the same comparing the B cell levels of a newborn, of a baby, of a children or child with 1 or 2 years than an old man with 80 years. Okay, so that's has a huge impact on the analysis, and it's very important to have that normal data in order to be able to perform a good diagnosis. So basically, by standardizing all of these panels, you can actually make sure that different labs are providing samples in order to be able to obtain this kind of data, which is very important. So because primary deficiencies are not that common, and luckily they are not that frequent, it's necessary that this data can be shared between different centers and among different laboratories in order to be able to obtain insightful insightful data from it and conclusions. So yeah, here we have again the same kind of information that we discussed in the first publication. This is the one that contains all of these papers, and this was in 2019, so 2 years ago. But actually, this is leading to more and more research. So, for example, in this very recent publication, 1 year later, in March 2020, they are analyzing the application of the EuroFlow diagnose, like the EuroFlow immunology approach to some severe PIDs in newborns and young children. So basically, they are validating how the approach that they designed before is feasible and what's the outcome of that in the clinical practice. And an important part that I forgot to mention is actually that all of these databases that EuroFlow is actually building, they are being included in Infinicyt. So when you run a sample in Infinicyt with the primary Infinicyt Orientation Tube, on the one hand, Infinicyt will automatically analyze the sample because of the automatic gating and identification. So Infinicyt will take that sample and it will process it, and it will give you a final result saying these cells are lymphocytes, these are B cells, these cells are T cells, these are T cells from this type, and so on. Not only that, but actually it will give you a report with comments and conclusions. And the point is that this result that you obtain, the frequency that you obtain, will be compared to the database corresponding to the age of the patient that you included. So when you run the sample, you can say, this is a patient with 16 years old and 5 months. So that will allow the software to use the right segment, and then that segment will be used for the comparison with your sample. So then, again, because it's not the same that this sample comes from a baby that if it comes from a old man, right? Okay. So yeah, here you have information about the different panels, more information about the application of these panels, and they didn't do that only for the primary efficiency orientation tube, so for the basic tube, because actually they did it for other diseases too. So for example, here they are analyzing the defects in memory B cells and plasma cells, and how they have different immunoglobulin levels, and how that affect how is that impacted or changed under different deficiencies. And I was looking for a different paper. Just a second. Yeah, I was trying to find this one. So this is also one of the initial publications related to the immunology research, and here they actually published the levels of the different cells, plasma cells, memory B cells, and the plasma immunoglobulin levels, depending on the age of the donor. Again, quite relevant data, providing a lot of information that will be able to compare with patient samples, and finally, so that we can actually assess whether there is a change between in the frequencies of the patient. So in this case, they are talking about basically this the pregerminal center and postgerminal center tube. So I'm just coming here to this to show you the panel, which is here. So here you have information of the panel, and then basically with this panel they are able to find all of that cell populations, and by assessing hundreds of samples they can find that they can obtain that numbers regarding the expression of the different immunoglobulins and the different cell populations depending on the age of the patient. As I said, all of this information is available in our website. So if you want to know more about the different disease categories, you can go here, for example, to primary immunodeficiencies, and there at the bottom we have some references, including all of those. So for example, the one that I show you, the primary efficiency orientation tube, the whole immunology panels, the one that I showed you, just the last two, including the levels of immunoglobulin, how they selected the clones of these immunoglobulin antibodies. These are available in our catalog, and they are working very nice. So if you are working with this, I recommend you take a look at those, at these immunoglobulin antibodies. And also important to note, just a few weeks ago, we released these two new papers or posters, sorry, these two new posters, including the EuroFlow panels. Okay, so this is the EuroFlow. You see that it has a similar structure to the EuroFlow immunology panels. So the PID there in the center, and then the other panels. So here you have access to all the antibodies and the programs used, and also some of the information required to do the diagnosis. This is for primary deficiencies. And then we have the hematology panels, including both the first phase and the second one. So screening, characterization and classification, and monitoring. This can be downloaded from the ESCCA section of our website, as I showed you later. Okay, so here in the brochures available, if you fill in this information, you can download here the two posters. Okay, so it's 2 minutes to 5. I wouldn't like to take much longer. I will drink some water. And basically that means that it's the end of today's session. I hope that you enjoyed it. If you have any comment or question, please let me know, and we'll be glad to talk to you more about this, or to solve any doubt or question that you might have. Tomorrow we are starting again at 10 with Cytognos about a session, just introducing the company and so on. And later we'll continue with all the sessions. Reminder, everything will be recorded and available in the coming days, and also in our website, hopefully. And yeah, that's everything from our side. Thank you so much for being here, and we hope to see you tomorrow. Have a nice day. Bye-bye.